Good morning, everyone. For ISGO2020, welcome to the third International Symposium on Germplasm of Ornamentals, and welcome to the beautiful city Incheon, Korea. I'm so glad that you are here with us today, especially in this difficult season with COVID-19. I'm very excited to have many of our participants in online. I hope that this conference today will be a special time for each of us, as we have participants who are joining us in this auditorium, and for those of you at your office or at home with the live streaming service. I just want to say a great big welcome to all of you. Well, to be sure, for a couple of things to help technical issues go as smooth as possible today. We are on YouTube and we are on Dockcast for Chinese. If we are having any technical issues where the server seems to go down or the screen freezes, the first thing to always do is just trying a quick refresh on your web browser that you are watching from. However, if that does not work, please jump on over to our webpage. We will try to let you know we are having some issues, and we will keep you updated on what's happening. But I hope everything goes smoothly today. Now I hereby declare open the third International Symposium on Germplasm of Ornamental. My name is Yoonjin Kim, General Secretary of the Organizing Committee. Since this is the first hybrid ISHS symposium, where the symposium is being held both online and offline, I'm very honored to be here to welcome all of you. This morning, we will have a welcome address by Byung-Yong Jung, convener of the symposium, as well as president of the Korean Society for Horticultural Science. After that, we will have a welcome message and ISHS presentation by Professor Cesar Kumar Mitra, ISHS board member. And then there will be a welcome message by Jung-ho Lee with video clip, representative from Korea National Arboretum. And there will be a keynote speech. Group photo is scheduled after session 1. Now, we will have a welcome address from Dr. Byung-Yong Jung, symposium convener. Please give him a big applause.
Everybody, could you turn your smartphone screen on for me? Thank you very much. Could you turn the lights on now? Good evening, America. Good afternoon, Europe and Africa. And very good morning, Asia. Dear Honorable ISGO 2020 participants and guests, we are finally here at the moment of opening the third International Symposium on Germplasm of Ornamentals in Incheon, Korea. And it is my great pleasure to welcome you all to presumably the first ISHS Symposium of 2020 and the first hybrid ISHS symposium ever. We have excellent keynotes, invited, oral, and post speakers ready to go. Now, I just hope that everything we put together for today and tomorrow runs very smoothly. I am so grateful for all the support and the help from you all who made it here and also who are unfortunately away but still connected by internet. I am indebted to the ISHS committee members, chairs, especially sponsors. We see on your right, and secretaries, editors for making the ISGO possible, including the publication of Acta Horticulturae in time. Finally, ladies and gentlemen, let's rekindle ISHS with the digital embers, and let's continue horticulturing with the multicasting connectivity. Stay safe and healthy, horticulturist. Until then, thank you very much.
It was a wonderful welcome address from convener Byung Yong-jung. Thank you very much. Now we have Zoom connection with Professor Cesar Kumar Mitra, ISHS board member, for welcome message and welcome ISHS presentation. But I would like to check his connection first. I think he was on Zoom 5 minutes ago, but there are some technical issues. So instead of his speech, I would like to have the welcome address by Jung-ho Lee first. Jung-ho Lee. Oh. So we will have welcome message by Jung-ho Lee, representative from Korea National Arboretum.
I'm Jung-ho, Acting Director of Korea National Arboretum. It's very nice to see you all. Congratulations on the opening of the third ISGO 2020 in Seoul, despite the worldwide pandemic COVID-19. Today's symposium is among a variety of subdivisions of the ISHS. It is disappointing that we are not able to join in person with many researches across the globe in this past season of our year fall. During this time, picturesque fall foliage emblazons Korea and crops ripen in fields. However, I am also glad that we are able to have online and offline symposiums at the same time. And I welcome you all who are taking part in via the internet. Ever since the official opening of the Korea National Arboretum in 1999, we have been engaged in various roles regarding plants and the ecosystem. As we spearhead the research on forest biospecies in Korea, we have pioneered a wide range of areas, including investigating, collecting, classifying of forest species, preserving, and researching rare and endemic plants, and promoting cooperation with arboretum home and abroad. Research topics such as promote the garden and wildflower industry and increase the value of Korean indigenous plants as ornamental plants are constantly researched in the arboretum. During this symposium, potential Korea ornamental plants that are currently researched will be one of the topics for oral and poster presentations. We have a lot of intriguing topics in this symposium, such as new ornamental plants and breeding, ornamental exploration and utilization, germplasm, resources and conservation, application of modern technology, researchers from home and abroad, and graduate students who have much potential to be great scientists. I hope you enjoy this symposium. Again, congratulations on the third International Symposium on Germplasm of Ornamental. I appreciate all of you who are present and participating in the event. Thank you.
Thank you, Jung-ho Lee. It was the welcome message by Jung-ho Lee, representative from Korea National Arboretum. We were having Professor Mitra, ISHS board member, on our Zoom connection before, but because of the unstable connection, maybe we can a little bit wait for him to have the welcome message or not. But instead of him, our convener, Professor Byung-Yong Jung, now he's preparing for ISHS presentation instead of Professor Mitra.
Thank you very much for kind introduction. Originally, the leaders at the ISHS, they were going to support this part and also the business meeting part. But it seems like we have some technical issues. So I'll just go ahead and present representing ISHS. Every symposium, we have representation coming from the ISHS headquarters and give this talk. And this time, since nobody could come, I was asked at the beginning to play the role. But since they were going to connect it, I asked them to do the job. But anyway, International Horticultural Society, abbreviated by ISHS, is a society of individuals, organizations, and governmental agencies devoted to horticultural research, education, and industry to improve human well-being. Dating from 1864 and formally constituted in 1959, the ISHS builds and maintains human knowledge and skills and biological resources in support of worldwide horticulture. And ISHS provides, through meetings and publications, both context and ongoing support for professional decision-making. And it explores and explains the many contributions of horticulture to healthy human environment. The ISH has a membership, thousands of individual members in over 150 countries, There are country and region memberships and also institutional membership. As you come into this room, you probably received one of these brochures. This is the application, membership application form. You can take a look. And the ISHS runs scientific programs such as symposia, like 45 symposia each year. And you can visit ishs.org calendar for specifics. International Horticultural Congress is held every 4 years. And in 2006, it was held in Seoul, Korea. The last one was in Istanbul, Turkey in 2018. And the next one will be 2 years in Oze, France, then Kyoto, Japan. The International Horticulture Congress in France is going to be held from August 14 to 20 in 2022. So if you are interested in, please save this date on your calendar. And the next one in Kyoto is August 23rd to 28th, 2026. The scientific structure of the ISHS has 14 divisions, including plant genetic resources and biotechnology, and ornamental plants, which our symposium is under this umbrella, I guess. And all symposia published in Acta Horticulturae, all Acta Cultura, more than 1,200, posted at www.actahort.org. The publication for this symposia is numbered 1291. It's already published. I think you already received one copy, one electronic copy. And there are more than 65,000 full-text articles there, and more than 160 library subscriptions worldwide, and more than 2,000 pages viewed per day, and it's a DOI cross-referenced. And ISHS have another publication called Chronic Horticulture, and it's published quarterly. It's ISHS membership magazine. So after we finish this symposia, we are also going to make a report to Chronic Horticulture. The next one is EJHS, peer-reviewed journal owned and published by the ISHS, and it's available in print and online open access format. And another one is Fruits. It's a peer-reviewed journal owned and published by the ISHS in cooperation with CRAD. Tropical and subtropical horticulture is the main format, range they cover. Okay, next. Another one is Scripta Horticulturae. It's a monograph published by ISHS in a series on special topics. This is the homepage you can visit for more information. And the address is www.ishs.org. And every symposium, we give award to young generation, best oral and post presenters, and you can find that information here, Young Minds. This symposium also, we are going to choose one best oral presenters plus one best poster presenters. Okay, another thing is probably many of you already have, is Harvesting the Sun, a profile of world horticulture. It's a publication some time ago. Pub Hort, it's a journal again. And ISHS have also Facebook pages you can visit using your smartphones. You are welcome to log on to the web page and apply for ISHS membership. Thank you very much.
It was a ISHS presentation from Professor Byung-Yong Jung. Thank you very much. Unfortunately, we don't have Professor Mitra connection for welcome message, but I'm sure that he would like to say welcome all of you for this first ISHS hybrid symposium. So now we will conclude the opening ceremony and move on to the keynote speech. The first keynote speaker this morning is Dr. Jong-seok Lee, Professor Emeritus of Seoul Women's University. We plan to explore native hosta species in Korea with his keynote speech. I am pleased to present Dr. Lee. Dr. Lee, could you please start the talk?
Good morning, ladies and gentlemen. It's nice to meet you. And welcome to attending the meeting of Third International Symposium on Germplasm Ornamentals held in Incheon, Korea. This is Jong-seok Lee. I am Emeritus Professor of Seoul Women's University and working at the Korea National Arboretum as a invited researcher. We are very sorry for the inconvenience caused by COVID-19. Who will stop this pandemic? Today, I'll be talking about one of Korean native plants. Exactly, my presenting topic is on Korean native hosta. Now, you are looking at the topic plant in front picture. And then I'll start my presentation from now on. Yes, do you know blank field, BBQ? Don't say bye-bye chu. This is Korean BBQ. This plant name is hosta in Korean. We are now very plentiful of this plant in our Korea. This BBQ is on Korean, maybe easy known, but maybe I think foreigners is not easy. Maybe this words would fit on this blank full. Maybe this one a little later, you can be able to fill up this blank, perhaps before my presentation over. Maybe this right word was blank is, I think, beautiful, or colorful, or graceful, or wonderful. Yes. Answer is yours. Choice is yours. I will give you next some hint. This is a hosta gardens in Korea. The Korean National Arboretum hosta garden. This hosta garden created by me 5 years ago. And the Goun Botanical Garden in 충청남도, and the 금원산 Arboretum in 경상남도, and the Jade Garden in 경기도, and the 한택 Botanical Garden in 경기도 near Seoul, and 무량기 Arboretum, this is in 경기도 오산. Next. Hosta garden in the world, left side of the upper line is Hanin Hosta Garden in Korea, and James Garden in Toronto, Canada, and the Chicago Botanical Garden in the Chicago. This is the Missouri Botanical Gardens in the United States in St. Louis, and the Toledo Botanical Garden, Toledo, Ohio. And this is a Hosta Garden of Indiana University at Bloomington, Indiana. This is a small gardens for using hostas. This is a Butchard Gardens in Canada. And the Sapporo personal home garden is in Hokkaido, Japan. And this one, Vladivostok, Russia. And Auckland in New Zealand, and Parnu in Estonia, and the Riga Public Garden and Latvia. This is a Krakow in Poland, a personal small garden using hosta. The Stockholm in Sweden. And this is a hosta garden of Ryerson University in Toronto, Canada. This one public park in Oslo, Norway. This one Bessel, West Germany, in the home garden. This is St. Louis, Missouri, and the United States of America. Okay. They are very colorful drip irrigation type of hosta cultivars. These are from Korean native species, all cultivars by my breeders. The white edge, the Korean name is white edge, and this is one origin of minor is vulture. Third one is May green, and Melusta origin is yellow dream, and the green wave is minor. Yunha is come from Benusta, and black liver is come from Hosta ingeri, and the sun and moon is come from Capitata, and snow line is come from Hosta clausa, and the yellow moon is come from Hosta capitata, and the bone is come from clausa, and the Hongdo is come from ingeri. Yes. Hosta cultivars bred in Europe and the United States of America. There's a Christmas candy, and the Francie, June, the Christmas tree, and the Liberty, Inniswood, there's Jones, Striptease, and the Legal Splendor, and the Fall Wind, Sun Power, and the Fragrant Bouquet. This fragrant bouquet is come from Hosta Plantaginia. And next. Next slide. This is fire and ice, and cherry berry, Shiro Capitan is come from Japan. June fever. This line is green margin green margin, the leaf variegation. And the blue dimples, the husband blue. This one is a blue color leaf. And the praying hands looks like praying, yes. The purple dwarf, this one come from Hosta venusta from Korea. Twilight and Independence and Stiletto. This is one white color. White color margin. Excuse me. Yeah, next. This is flowers of various hosta cultivars. Upper part on the left was on Big Daddy was on white color, and the blessing hand blue is white color, and the purple dwarf and the yellow splash means a deep purple color. They are various in color. Yes, introduction. This is hosta genus. I'm talking about the genus Hosta. Hosta is an Asparaceae since 2009. Before 2009, this plant is belong to the lily families. There are many kinds of hostas growing in all of Korea. And as you can see the front, this native hosta are using an important resources for breeding new cultivars. In Korea, about 300 cultivars are planted in the garden, and about 3,000 cultivars in the world nation's garden. Hostas are very hardy garden plants, so they can grow in the open air of northern hemisphere regions. For example, USA, Canada, Northern Europe, of course, Korea, Japan, and China. However, natural habitat of genus Hosta can be looking at East Asian regions only. Natural habitat in Korea, Japan, and Northwest China, and the Far East Russia. Russia indicating Kamchatka Peninsula. Especially a lot of hostas, natural habitat is easily found in Korean Peninsula and all of Japan. As you know, classification of the hosta species very flexible and confusing according to the taxonomists. For example, Maekawa recorded 39 species in Japan in 1940, and Fujita published 15 species in 1976, and Schmidt classified 43 species in 1991, and Jorebolt and Eden classified 24 species in 2001. However, the reality of the researchers in Korea is exactly the same as the former case. Now, I would like talking about the classified 6 Korean native hosta species that can be looking at the natural habitat. These are Hosta capitata, Hosta clausa, Hosta jonesi, and Hosta minor, Hosta venusta, and Hosta ingeri. How to searching the habitat? The habitat research was conducted as follows. At first, we reviewed the published important vegetation report from 1870 to 2017 during 105 years report. And searching the native place have been from April to early October. And the intensive investigation was conducted to flowering season from July to September. Yes, this picture is flowering view of a natural habitat of Hosta capitata on prairie of Mount Taebaek Mountain in Gangwon-do. On the right side, the green map is present the 6 vegetation limit in Korean Peninsula, Northern Province and the Middle Province and the Southern Province, the South Coast Province, and the Ulleung-do and the Jeju-do Province. This is our surveyed map here. This right side upper is on Vladivostok, located in East Russia, and Yanji regions, those Eastern China. Where is adjacent to the border of North Korea, because we cannot go North Korea's habitat. And we are mainly searching the southern part of the militarization zone in Korea. Militarization zone is from east to west, 155 miles. For example, Gangwon-do and this is Gyeongsang-북도, and this one is Jeollabuk-do, and Jeollanam-do, and Gyeongsang-namdo, and southern island of Jeollanam-do and Jeju-do. Yes, the left side picture is a flowering in bloom of capita in natural habitat of Gangwon-do. This picture is Hosta minor habitat in the woodland forest in Jeju-do. You can see growing appearance of a cluster in their natural habitat. On the right side, this is in Venusta on the steep, heavy woodland in mid Halla, Jeju-do, Korea. Maybe this flower will bloom soon. This photo is pictured on mid-September this year. Next. Yeah, I will explain the result of the research papers chronologically. Number 1 is 1870. Hosta minor was recorded at the first in the Journal of Linnaean Society by Baker as a new variety of Funkia ovata, spring variety minor. Baker's British researcher, he confirmed Hosta plant specimen collected from Korea by Richard Oldham in 1863. Number 2, 1901, Funkia lancifolia and the Funkia ovata. Funkia means ancient Hosta's name, family name, and collected from Amnok and Jangjin River, located in the northern area of Korean Peninsula. This plant was recorded by Russian Komarov on the Flora Manjurika. Number 3, 1911, Hosta rancifolia and Caerulea, the Hosta minor, were documented by Nagai on the Flora Koreana past second as a Korean native Hosta species. Number 4, 1922, Hosta Caerulea, Japonica variety rancifolia, and Longipes variety alba, and Siboldiana were documented by the Mori on the enumeration of plant hitherto known from Korea. This is a picture. This picture is appearance of Hosta Siboldiana, and another is Caerulea. There are no natural habitat in Korea, but in Japan. Number 5, 1913, 5 Nakayana collected from at Baegunsan located in Jeollanam-do, and with Jirisan located in Jeollanam-do and Jeollabuk-do area, was described by Maegawa and Smith as a new Hosta species in Korea. Number 6, 1937, Hosta japonica borea caerulea, japonica rancifolia, Longipes variety alba, Hosta minor, and the Siboldiana were recorded in the flora name of Joseon. Joseon means an ancient Korean name. By Korean plant taxonomist Jung Tae-hyun and his colleagues. And number 7, 1949, Hosta plantaginia and the Undulata variety aromena were documented on the name of a Korean plant by Bang Man-gyu, who is a Korean taxonomist. Hosta plant is native to China, and the Undulata aromena is native to Japan. This species cannot find a native place in Korea. Number 8 is Clausa variety normalis, and Japonica variety rancifolia, Japonica variety latifolia, and rancifolia and Longipes, the Hosta minor, were documented on the Flora Korea by Jung Tae-hyun. This picture is Hosta plantaginia, and the right side is Undulata variety aromena. These species are not Korean native species. Number 9, 1980, Hosta minor, Clausa normalis, the capitata, rancifolia, Longipes, and the Hosta japonica variety rancifolia were documented on the illustrated flora of Korea by Lee Chang-bo. And number 10, 1985, Hosta ingeri was recorded by Jones, and the new species of Hosta. These plants were collected from southern island of Korea Peninsula by Barry Inger, who was a Korean plant collector of USDA in US. He stayed about 4 or 5 years, stayed in Korea in 1980, early 1980, I guess. 1985, 11 species, 2 varieties, and 1 form of Korean native Hosta species were published by Jung Young-chul on the Korean Journal of Plant Taxonomy in 1988. 12. In 1985, as a new species from Korea, Hosta jonesi distributed in the southern seaside of Korean Peninsula were published on the annuals of the Missouri Botanical Garden in 1989 by 정명기. 정명기 is a professor of Gyeongsang University. 13, in 1991, Hosta venusta, Hosta minor, Ingeri, Jonesi, and Capitata, and Hosta clausa have been readjusted as a real Korean native Hosta species by Jung Myung-ki. And the number 14, in 1996, 5 species, 1 variety, and 3 forma were documented on the flora of Korea by Lee Young-no. Dr. Lee Young-no is an Ihwa Women's University professor. He's gone. Number 15, in 2006, 6 species, 1 variety, and 3 forma were duly documented on the flora of Korea by Lee Young-no. At last, 16, 2070, 4 species, 4 varieties, 6 forma, like this one, were readjusted on the Korean Journal of Plant Taxonomy by Cho and Kim. Yes, I will show you appearance of each Korean native species and explain their distribution and characteristics. Hosta capitata. This is first recorded in 1930 by Japanese scientist Takai. Korean name is 일월 비비추. English name is capitate plantain lily. That the distribution area is central and the southern region of Korea and the Shikoku and the Kyushu in Japan. The flowers bloom July to August. Number of floret 12 to 23, 2. Floret number is 23. The scale length 60 to 86 centimeters, having reaches longitudinally on the scale. Flower color is light purple with deep purple lines. As you see the flowers. Characteristics, the floret is clustered on the top of the scale. Variety is Hosta capitata, Horma albiflora. Albiflora flower color is pure milky white. This is the Hosta capitata, and the habitat of capitata in Gangwon-do. And the right side, Capitata forma albiflora. Next. Hosta clausa. Number is Josie clausa. There. Yeah. Clausa. First recorded the year 1930. Korean name is Jugok Pibichu. Jugok means spatula or sword. The leaf leaf shape looks like a spatula. And distributed mid to northern Korea and Jirin, Yoryong province in China, the northern part of Korean Peninsula. Flower is bloom mid to late August. Floret number is 14 to 15. Scale length is 73 centimeters long, no ridges, so these are smooth. The flower color is deep purple, and the characteristics close to flower. And so this is the no seed, triploid plants, and propagated by underground stolon. Variety is close variety normalis, and the difference is an open flower. The flowers are variety 금강gensis. This is from the opening is white color and no purple. Yes. Right side you can see the propagated underground stolon. You can see the lot of plantlet growing around the mother plant. Middle picture is middle, shape of open flower, and light variety of 금강gensis. Flower color is white. Okay. Next, Hosta Jonesy, first recorded in 1989 by 정명기. Korean name is 타도해 비비추. Distribution is the southern seaside areas of Korea, 거제도, 남해, in 경상남도 area. Flowers bloom August to September. Number of floret, 20 to 17. Characteristics is plant height, 30 centimeters high. Scape length, 73 centimeters. No ridges, so flower scape is smooth. Flower color is light purple with deep purple line. This is, now you are looking at the leaf shape of Hosta jonesy. The flower leaf color is whitish green surface color, and the wrinkles around the leaf edge. The floret hang down. Second means facing one direction only, and or subsequent at the upper part of the scale. Variety is Joraciforma albiflora. The differences is flower bloom in 5. Hosta minor, first recorded in 1911 by Nakai. Korean name 좀 비비추. Korean name 좀 means small. So small hosta. English name is Korean hosta. Distribution is mid to southern regions of Korea, from 강원도 to 제주도. Flower bloom June and July. Number of florets 22, 27 florets. Having ridges on the scape. Flower color is light purple with deep purple line. This plant is an endemic plant in Korea. This picture is a different type of leaves according to the native site or individuals. Flower color of purple tint is different to the individuals. The center is large leaf type, and the small foliage type is hosta minor is right side. Variety of hosta minor, flower color is white. You can see the white flower color. This Venusta is recorded 1935. Korea name is Halla Bibichu. Halla means mountain name in Jeju-do. So this area is this habitat. English name is dwarf lily, plantain lily, or maiden hosta. Distribution is mid-Halla in Jeju-do. Flowers bloom June to July. Number of florets is 6 to 7 florets. Plant height is 5 centimeters. And the scape length 16 to 20 centimeters. Longitudinal rigid, not smooth. Flower colors purple and pale blue, and the leaf dwarf and the smallest among the hosta species in the world. This plant is an endemic plant in Korea. Left side picture is close to vegetation in the natural habitat. And the middle is growing appearance on the steep, on the heavy woodland forest. Right side is appearance of a bloomed flower. Hosta ingeri, first recorded in 1989 by Jones. Korean name is Huksando Bibichu. Huksando means island name of Korea in southern part of Korean peninsula. And the English name is Ingeri hosta. And flowers bloom on August. A number of florets is 66 or sometimes over 100 florets. And the flower shape looks like a funnel. And the flowers turn around, and the petals are thin and looks like a spider. Characteristic is the plant height 28 centimeters, and the scape length 82 centimeters long. No ridges, so smooth. Flower colors light purple, leaves wide and gross. This plant is an endemic plant in Korea. Yes. Yes. Hosta ingeri, leaf is wide and gross in the right side and left side, and the shape of the flowers and the scape and the hosta levigata, WG, Schmidt. However, I guess that type is maybe a control variegation. Sometimes I could found this type of plant among the seed propagated plantlets. Okay. Yeah, finally, as you can see the table, we suggest the classification keynote of 6 Korean native hosta species. I'll explain the tables. Number 1 is blacks, papillus at apex, and greenish when fresh, remaining green at flowering and persistent with the fruit. This type is Venusta, minor, and Ingeri genus. But another type is bracted not papillus at apex. The fading to whitish brown at flowering, not persistent with the fruit. This type is on Hosta capitata, Hosta clausa. And number 2, this one. Scape rigid or longitudinally straight. Another one is scape smooth, not rigid. A scape rigid type is Nebusta and the minor, and scape smooth type is Ingari and the Jonesy. And Nebusta is leaf is small. Hosta minor is a leaf broad, large. This is different. And number 4, Ingeri is disticus stamen. And Jonesy is a secund, also secund. The length of a stamen, the same. And answer whitish and whitish yellow. Rhizome is underground stolon. This type is an Hosta capitata. And answer dark purple. Rhizome is underground stolon. This type is an Hosta clausa. This is a Korean native hostas classification keynote. Very important keynote. Conclusions. History of Korean native hosta has been made by Baker, 1870 at first. And the number of Korean hosta, native hosta species was confirmed as 6 species, 2 varieties, 3 forma by habitat observation throughout Korean peninsula. 6 native species is Hosta capitata, clausa, Jonesy, and Venusta, minor, and Ingeri. 2 variety is Clausa variety normalis and the Clausa variety Kumgangensis. 3 forma is Capitata forma albiflora, Jonesy forma albiflora, and minor forma alba. Leaf size and the shape of minor was different depending on the habitat and the individuals. We suggested that classification keynote of a Korean native hosta species. Yeah. Yeah. Yeah, thank you very much.
Thank you very much, Dr. Lee, for your excellent presentation. For the participants on Zoom, I would like to inform you about some technical things. I'd like to suggest you to watch YouTube or Doccast for live presentation. You can talk and you can hear via Zoom. But please remember that you can check other presenters' talk on YouTube or Doccast. It means you can use Zoom only whenever you would like to talk for the question and answer, and also for the group photo time before the lunch. So now, before we move on to session 1, the Professor Cesar Kumar Mitra sent us his welcome address by instant email. Unfortunately, we don't have him on our Zoom, but our convener will read that instead of Professor Cesar Kumar Mitra, ISHS board member. Could you please start the talk when you are ready?
I'll read Dr. Mitra's message, welcoming, congratulatory address. He was going to be online, but there were some technical issues, and he sent me email in the meantime. Welcome address. Distinguished delegates attending on-site and also online from different countries of the world. Professor Yoonjin Kim, Secretary General, Professor Byung-Ryong Jung, Working Group Chair, Ornamental Genetic Resources, ISHS, and the convener of the third International Symposium on Germplasm of Ornamentals, Professor George Kanhoto, Chair, Plant Genetic Resources and Biotechnology, ISHS. Ladies and gentlemen, I, on behalf of the President ISHS, board members, and the commission chairs, welcome you all in this symposium. 12 months ago, I doubt any of us could have anticipated the different world we would be living in now. COVID-19 has spread around the world over many months and continues to cause havoc and the losses of life and severely restrict international travel. For ISHS and its members, it has resulted in the postponement of many symposia and, in few cases, cancellation. This has caused us to accept that we, as a society, need to adopt a different strategy. We now encourage symposia conveners to prepare for full online or hybrid symposia. I'm happy that you are successfully organizing the hybrid symposium, and participants, those who are able to travel, are present here at the venue, while those unable to travel are attending online. Many national societies have already organized very successful online conferences. For ISHS, however, we have the added challenge of delegates from all time zones around the world. I'm pleased to see that you have already published Acta Horticulturae out of this symposium and the volume 1291, which is more than 300 pages containing 35 full papers. I would like to place a record and thank all the editors, Drs. Wansung Kim, Seungyeon Lee, and Yonghao Lee, for their hard work, even during this pandemic, to make it possible to release the ACTA during the symposium. I understand that you will discuss germplasm resources and conservation, new ornamentals and breeding, exploration and utilization, and application of modern technologies of several ornamental crops like veronica, hydrangea, chrysanthemum, rose, fern, corydalis, oriental lily, azalea, angelica, poinsettia, and many other ornamentals in the coming 2 days. And I firmly believe you will come out with recommendations for conservation and utilization of ornamental germplasms. On behalf of the ISHS, I wish you all the success of the symposium and look forward to meeting you in person at the next symposium. Thank you all. Professor Dr. Cecile Mitra, board member, ISHS, responsible for publication, dated 26 October 2020. Thank you very much.
Thank you very much, Dr. Jung, to give us one message from Professor Mitra. And thank you very much, Professor Mitra, to sending us this wonderful welcome address. Now, I would like to turn the chair over to Dr. Ki-sung Kim from Seoul National University for the following session 1.
Good morning, good afternoon, and probably good night in some region of the world. This is my honor to chair the first session of this international symposium this morning. I have 7 speakers, 2 invited speakers, and 5 oral presentation, and 1 from United States, 4 from China, and 2 from Korea. First, we have invited the speaker. Let me introduce Dr. Donglin Chang, my very old friends for more than 20 years. Dr. Donglin Chang is a professor at the University of Georgia, USA, and the prime investigator of University of Georgia, Woody Plant Research Laboratory. He earned his PhD from the University of Georgia and worked as an assistant, associate, and full professor at the University of Maine, with focus on cold hardy, ornamental, and horticultural plants. Dr. Zhang also worked at Harvard University as a Putnam Fellow for woody ornamental plant physiology using living ornamental plant collection of Arnold Arboretum. 4 years ago, Dr. Zhang organized and successfully hosted ISGO 2016 in Atlanta, Georgia. He's very active at the International Society for Horticultural Science and American Society for Horticultural Sciences, and focuses on sharing Asian horticultural plants to our colleagues around the world. With many professional visits to Asian countries and 3 symposia on Asian horticultural plants, he greatly promoted Asian horticulture and significantly enhanced Asian horticultural sciences in the world stage. Dr. Zhang's laboratory focuses on, first, ornamental germplasm exploration and collection in North American, Southeastern Asia, and other similar natural condition regions around the world. The second, breeding and selection of new woody ornamental plants. And third, appreciations of modern embryogenesis and DNA technology for plant breeding. His lab has released more than 20 new ornamental cultivars in the past 8 years and published 2 through 7 references papers per year. Very productive. Today, he would like to share with you some research findings from his lab. Since we are moving early than on time, I'd like to, Dr. Zhang, take your time, and you have 25 minutes talk, but you can have some more if you want. So, Dr. Zhang, are you online? Oh, there.
Yes.
Hi.
Yes. Thank you very much. Thank you, Dr. Kim, and I'm really glad to be here with all of you and today and share some work. But actually, I would like to share with you, how to do woody ornamental plants, especially when the technology developed so much. We have to borrow some knowledge to share with people. So today I will share a little bit what my lab did, and also I want to encourage you guys join us to do more ornamentals. Since I have more time, I will not worry to hurry in 30 minutes. So I have many people in the lab. I selected Dr. Yu Jie Yang as a co-author because I just want to use her as a representative. I really like to thank the convener and Dr. Pei-Yong Chong and all others, especially the International Society for Horticultural Science, Korea Society for Floricultural Science and Horticultural Science, Those symposiums take a lot of time and energy. I really appreciate they're doing so wonderful job. They already have proceedings published. I remember I hosted 3 of them, and they usually take 1 or 2 years to publish the ACTA, but they did a very, very good job. I appreciate and thank you very much for giving us the opportunity to get together. Talk about woody plants. We all probably know any garden without woody plants is not really the garden. So it is a backbone that make a lot of structure, make a lot of design work, and also make your garden have the layers of the observation, the landscape. So you can say here in a garden without the woody plants, it's probably very hard to have anything. Also, you can use woody plants for hedging, for screening, for what? For multiple layer planting. So I don't want to go too much. Woody plants is very important, not just on your garden, also on the forestry, our living expenses. Think about this world without a tree or shrubs. It's impossible for us to live in with these conditions. Now, some of you said, gosh, we have very little ornamental plants in the world, especially like, for example, crab apple. When I visit those gardens, which is in the Ohio, US, the fantastic diversity of ornamental crab apple is just amazing. So when you see this kind of picture, anybody ask in your mind, how the world, you get so many new plants, the diversity of plants, the beautiful plants, include shapes, flower color, and everything. So this is the kind of stuff you probably keep in mind, what's going on, how we do the ornamental plants breeding. So if I want to start it, how can I do it? So this is the major message I want to deliver to you today is develop a breeding program in general, like any other program, right? It's like a pipeline, like you manufacture of something. The number one issue is you have to collect it all possible breeding line, which is germplasm, as many as possible, right? Well, and now we have a new term called new genetics, new genes. So when you get all genes together, you use appropriately breeding selection technique. Those technique will be use a tool that you generate, right? Generate some new cultivars. So generally speaking, this is what we try to do, right? This is you can do the same thing, right? Don't forget, if you don't have germplasm, you cannot do anything, right? You cannot go on. Where's the germplasm from? From wild, from the collections, from nature. The nature produces so much. Now, what is the challenging part, right? When you do those ornamental breeding, it's very expensive. It's not cheap, right? And also, you need have big amount of funding to start it. Now, I don't know the Asia countries, but in US, the labor cost is tremendous, right? Tremendous. So this is what the major. But actually, when you do some breeding work, you need a lot of people. You need a lot of room and labor force to carry out the work. Otherwise, it's very hard, right? So don't forget, if you want to be successful, do anything, be dedicated, right? Be dedicated. Not just think about doing it, you want to do it efficiently. This is my advice to you. Make sure you keep in mind. Those advice will not stop you, hopefully. Hopefully you can continue to do this work because the only things you do, just keep forward. Don't step back when you have questions. Face the questions, anything you meet. you face it and try to solve it, try to gain your territory, right? You have to think about time requirement, scheduling. For example, if you do want to cross hybrid, you have to make sure you do understand woody plants, majority of them, especially in the temperate area, they flower once a year. If you miss this window, you missed it, right? Also, you need to get test locations, right? Test the design. You are not just simple amateur breeder. You are a scientist. You have to make sure what you do is good for the scientific area. And when you send the paper, they have scientifically sound to accept it. This is a very important, right? Here, what do you do? Everybody knows the Laxstromia, the crepe myrtle, is a big tree, huge. What do you think about those, right? I have run a huge program on the dwarf Laxstroma gradient. If you look at those diversity and the planting, you can imagine how much time, energy, knowledge, labor, money you need to put it in. This is an amazing thing. When I get this photo, I'm just so surprised because it is so showy, so beautiful. It's amazing. It's amazing. So think about those. You need to do step by step. If I were you, I don't have those luxury for the land. We don't have luxury for the money. What do? You do small section, do small part. So don't be scared. Another challenge I want to pose to you. If you think about your work on the rice, you work on the soybeans, you work on the big flowers, how many people work on it? Thousands of people. If you work on the ornamental plants, how many people work on it? You're maybe the only one, right? Especially when I go talk, when people ask, you're the breeder, what kind of plants you're breeding? I always tell people I have many of the plants because on the woody plants, right, you have to do many species, not just one, right? This is a very, if you work on some herbaceous plants, some annual plants, you can grow in 1 to 3 generations per year because we can manipulate. For example, now we growing vegetables, we could growing 2 or 3 times a year. But for the woody plants, you can't 1 time a year. This is very important. And also you have to learn everything about the plants by yourself because no other people is available do this kind of work for you, right? So do you still want to do the plant breeding? You probably kind of hesitate, but I can tell you, it is fun, it is very enjoyment, and it's rewarding. Why? Because you can use modern technology, right, to help you better breeding ornamental plants. This is one of the key issues. My lab at the University of Georgia separated into 2 parts, right? One part is research farm, another one is a laboratory. And the research farm is majorly in the field. You can test the trial greenhouses and the planting. This is probably you can say a little bit. Also, the lab crew is very, very good in terms of those. Now, inside of the lab, we do some seed technology, we do tissue culture, we do DNA technique, we also do some embryogenesis. So all of those combine and make the things works. So I will give you some example. It's very important. Now, as I told you at the beginning, when you start your breeding program, what do you need to do? First things, you need to go wild, to go anywhere. Your garden, your friend's garden, your university garden, your leisure arboretum, whatever you can, put your hands on it, collect all the possible germplasm as you can, right? We went to Southeast Asia because it's very similar to the climate here in the United States. And in the wood, it's like kids in the candy store, like heaven for us, because we love the plants. We can walk those days and days without any problem. They're so rich. the storage. The only thing is there's so many plants in the wild we have not known yet. I do encourage all of you to try to go there. It's not easy. You need to prepare yourself, right? Here is the delegation to the Vietnam. We have 2 from Canada and 3 from UK and 4 of us from the United States, right? All of those get together. We try to explore this area. The gentleman in the blood, and he went there for 20 years. Every year, spend a month there. It's just amazingly rich floral area. We just so blessed to see those plants. And make sure you do understand, we have many dangerous stories. Even one of person from Scotland, he went in by himself. While we walk out, he never come back. He never come back to the world. 2 years later, his body was found on the top. But anyway, be careful and go together. Don't by yourself, right? But you still have a lot of potential there. Now, when you get all those germplasm, what do? You come back and grow it. For example, this is a seedling of sky pencil. From the seedlings, what we do is we grow a bunch of them. As you probably noticed, the sky pencil is a beautiful plant. A lot of people work on it. But when they're growing bigger and older, what happened? The branch fell down, right? So the branch fell down. Those is the kind of challenge. What we try to do is how we avoid this problem. Then what we do, we take the seedlings, right? We growing those in fertile. So I will talk about those. We use embryo rescue to do that. How many of you know the ilex holly germination take at least 2 to 3 years? So if you're graduate students, you come to your program, collect the seed for 1 year, then wait for the germination for 2 or 3 years. What happened? 3 years is passed. You have not get any data. You want to graduate, right? Can you do that? You can't. So what we try to do is we try to cross it and then take immature embryo, right? Then from immature embryo germination, we can get those seedlings in the 1 year, So this is probably just show you. When you do this, there are many challenges. When you do the embryo rescue, embryo germination, the challenge is when I should take the seed, the immature embryo. For example, here, give you some ideas, right? I cannot say those. If you look at those, the contamination rate, germination is higher, contamination is also higher, depends on when do you take it. So number one issue, you have to make sure you have to try it every week. So make sure you try every week. You can do that. Then what do? You have to make sure which media is better for your seed to germinate it. Those data is all published because we tried many different. You can look at it. Then finally, your shoot growth. You make sure you promote the shoot growth. And those are steps for you to think about it. Time to take immature embryo, the media for germination, and the hormone for plant growth. So this is what you need to think, right? It's very important. I will not go more details because you guys can do this. You understand it better. So it's very important for you guys to do that. Now, compare with this this is when they're young, it's very beautiful. When they get old, the branch flop. Now we get this one, right, in the market now. This year, Bailey is marketing this plant called Chubby Hobby, right? Is in the market. That's what we do. Now, the key issue is immature embryo germination and the development. Is it easy or hard? Sometimes it's easy, sometimes it's hard. Colmea is like a rhododendron. The seed is very, very small I have a PhD students who work on those. Instead, sterilize the seed, the immature embryo. We have to sterilize the whole fruit. From whole fruit, then we take the immature embryo, then germinate it. This is why sometimes life is a lot easy. When you meet the tiny, tiny embryo, you barely can see the seed. How do you see the embryo? This is probably more important, is very important for you to think about how to deal with this. Now, the key issue is if you want a breeding of plants, don't forget what your goal, what the purpose, what kind of plant material you expect after breeding. For example, when I come here, viburnum is one of those plants. The local people really want the compact. They don't want the leggy, right? The ornamental plant. They want evergreen, so that way in the winter, they still have some plants to say. They also want cold hardness. So I don't want to say in North America, the climate change, especially the day and the night temperature fluctuation, sometimes is about 60 Fahrenheit degrees. It's amazing. Plant is very hot. Then you want the fragrance and the flower color. With all those goals in mind, you begin to what? You select the Viburnum carlisii, which can be grown in Korea, North China, and also in part of Canada. So they're cold hardy. Also can grow in here in Georgia too. Then what we do, we have a lot of plants which is called prolific. It's an evergreen hybrid plant. So what we did is we cross. Again, viburnum is also a lot of plants may take 2 or 3 years to germinate it. Remember that. You cannot wait. If you cross half its seed for 1 year, germinate for 2 years, doesn't make you work on the master, PhD, or your assistant professor, you want to get promoted. This will not give you anything to, right, to publish, to produce it. Now, we have to think about any way we can use the modern technology to help us, right? This is what we did, right? We crossed those 2 years ago in March 27th, 2018, right? This is probably 3 years ago. Now it's really time is so fast. When we crossed it, we then take the immature embryo on June 21st. This is about 86 days, 3 months later. Guess what? In September, another 3 months later, your seedling have 2 pairs of true leaf. So instead of 3 years, how much time you spend on it? You spend a year. Now, this is one part. Second part is, we use all of those if you design the experiment right. What do? You can publish the paper, which can help you to get your degree, get you tenured, get you promoted, right? If you're a researcher. This is what I try to deliver is, we not just simping. We not just step back by the difficulty. What we do, we need to think about smart, think about intelligently, try to use the modern technology to help us out. Otherwise, you just wait there. Now, if you have the good background on DNA markers, what do? You can take this seedlings, extract the DNA, and then compare it. So this is what happened. When you compare it, what do? You can figure out whether they are true hybrid or not, or they have any potential. If you can do some trade-associated markers, you can figure out some trait they are hybrid or not. So this is all together can help your publication, right? Can help your publication. This is what I think is you have to think about, use those. Anytime, doesn't matter your assistant professor, your professor, or students, graduate students, what do? you try to challenge yourself, use your brain to advance your knowledge, right? This means hard work and dedication. Hopefully everybody's ready. Now, instead of publish and do the research work on those embryo rescue, what do? Sometimes you don't know what you found. For example, when we work on the viburnum setigerum, right, it's a lot of viburnum, which is a beautiful ornamental plant. Guess what? When we take each immature embryo, do you know how many seedlings come out? What does this mean to you? So what I try to make a point is, if you do it right or do it carefully, sometimes you find some surprise, because this plant is what? It showed up as what happened? This plant is showed up as polyembryonic plants. So some of the states in Georgia, in United States, treat this plant as invasive, but they don't know why they're invasive. Now you understand that when you have one seedling germinated, how many seedlings come out? I can tell you, there's so many, right? So there's so many seedlings come out. Here is it. So if you say 2 to 3 embryos per seed, it's probably majorly 65% is 4 to 6, some even to 7 to 9 embryos. In other words, each seed you germinated, you expect how many seedlings? Right? You expect 2 to 9 seedlings. Now, what does this mean to you? Good or bad? If you're a breeder, you need to make sure, be careful. All those 2 to 9 seedlings, only one is embryo. Others is from what? Is from apomixis. Apomixis is another phenomena, which is you have to be careful about those. If you do the germination, you're all set, right? You can clonally propagate this plant by so many, right? By so many of those, which is very nice. Very nice. So this is the important part. You guys have to make sure you do understand those. You do understand those. Now, what I try to make a point is, when you do the research, always keep your mind open. If you find some potential and growth, you can always do it. Make sure you can publish. You can publish. Now, a lot of technology we want to engage is DNA. I'm not expert of those, but we can borrow the technology today to use for our work, right? So it's generally, I don't want to go too much. We get the seed, the immature embryo germinated seedlings. You can take a piece of leaf and then try to identify if there have any difference, any potential. So this is what we try to do. I don't want to go through those procedures, but at least our lab usually use ISSR, which is a very simple, easy one, right? You can say, by comparing all of those, we can figure out whether the seedling is worth to save it, is worth to use it, or it is nothing. So I will talk about more example later, about hours later, to share with you about more about those. So DNA technologies, they have so much application we can use, right? Like ISSR, FOP, SLAPs, GWAS, we can use all of those. And recently, we have a visiting professor from Beijing Forestry University. He initiated a CRISPR-Cas9 technology to do the hydrangea, which is, this technology is awarded Nobel Prize this year. So it's very important. We are not the genetic expert, but what we can do, we can borrow their technology to use for us. This is why I do encourage you guys to do the same thing. Not just think about woody plant breeding. You need to think about outside of the box. Begin to borrow more technology to do that, right? You use those technology to do what? Hybrid delineation, discrimination. So what you try to do is you can use this technology, not just screening. You can do identify is a hybrid or not, right? Especially if you can do morphological treat associated with marker, you're even better. So those technology is take time and energy too, but we don't need to invent ourselves. We can borrow up. The key issue is what? You speed up your grading procedures. Don't think about woody plants sometimes take 30 years, but now you can use those time while work on it and publish the paper, keep your survival, and then right advanced. When you get to the stage of a professor, you don't worry about it. You just work on it, right? You have so many things started, yeah, better. So this is why when you get a full professor, you can work on it. Guess what? When you get on the stage, you never go back. You will continue to work on it. You will continue to work on it. So this is why they don't care about give you tenure. And give you tenure is what? Give you the confidence to work on the same project forever, right? They know you're not lazy. So this is probably, don't forget, think about time and the labor cost, right? I have presented those rapid woody plant breeding system long time ago. So I will probably repeat the goals there to say, remember, when you crossbreeding, you make sure you, number one, you need to pick up all the possible, right? Gene plasma is possible. And those gene plasma, then you select it, say which one, you can use DNA technology to figure out which one have more genetically distance. So which one cross can be make better. So you have to figure out those. Then after you cross it, don't wait. The plants mature, collect seed, germinate it, right? You can do that, but you can use embryogenesis, embryo rescue to do that. One part embryogenesis is good is if you find a new plant, you can produce it what? Zillions, right? Very quick. If you use embryo rescue, you can save you at least 2 to 3 years, and some publications too. Those one give you what? Give you degree, give you promotion, give you security of your position, right? Then from those seedlings, you need make sure to check out, use DNA technology, right? So this DNA technology you can work on from what? From your gene plasma collections, right? So when you collect it, explore collecting all the possible gene plasma, you can try to use the DNA technology to work on the trait associated markers. So when you get seedlings, you can cross-check and make sure the mark trait analysis give you some more information. After you get all of those, you can initiate screening your selection, right? Those initiation of selections can also help you to do what? To do lease trial in the field. Then you have to grow in the field, make sure they're doing well for your area. So hopefully the industry like it, the industry will like the market, and then you get some royalty return to continue your work. This is how this works. And this is what I want to share with you today, and also encourage you to not scared by the woody plant breeding take too long. The important part is what? You try to use your technology, use your brain, and you all smarter than us, right? Especially the young generation. You should be able to do much better job than us. I hopefully you encourage, hopefully to see more young generation join us to do the same work, right? Now, I want to thank you, all this lab crew. You know, the people come to my lab, try to work, they contribute. all of those. I cannot put all of them on the name, but I really, really appreciate all the contribution they're made, right? They're made and make those talk possible. So thank you so much. And woody plant breeding is a challenge, but I can tell you, we can do a great job, especially when we work together. So as Dr. Ken Sang Kim told us, we know each other for more than 20 years because we always face each other. We have too much to talk together, right? So I visited Japan, Korea, China many, many times. For what purpose? For one purpose, share the information, right? Any people come here, if you want some plants for free to take it, we never close our door. Anybody wants plants, we want you. So welcome. Hopefully you guys will join us for ornamental breeding and especially woody plants. And hopefully someday we can work together and breed many beautiful plants to our garden. Thank you. If you have any question, I would like entertain you. Dr. Kim.
As you saw, he is always energetic and very greedy for research, even though he's getting old like me. He spoke about many things, from the field exploration, hybridization, and some screening, even to DNA analysis. So from conventional to the modern techniques, thank you very much for your great presentation. So do you have any question in the floor or by internet? Anybody in the floor, you can take a microphone, or internet, you can log on. Any question? Let me have a quick question before the people ask you a question. You talk about the Ilex crenata hybridization, and you saved many years to make seedlings. Conventionally, it used to be 3 to 4 years. Now you have only 7 months to have two true leaf, the seedlings. How about the flowers? Did you see the flowers from the seedlings?
Well, the flowers take at least 2 more years.
Yes.
Because this one you cannot speed up. And you have to plant in the field. They are for some reason, they're very quick. This is a surprise. We just get the flower, pink flower, last year. It's amazing. It's amazing. So you can save those first part, but the second part in the field is all depends on the plants. Some plants take longer, take shorter. We don't know how to speed up those. We need to think about it. Someday, some audience will answer this question. I can't.
Yeah, hopefully you can find some the techniques to shorten the period for flowering. Then it will be much more productive findings. Okay, any questions? This is a great presentation. So it was so clear that everybody understands what you're talking about. Okay, then if you have a question, you can personally contact Dr. Donglin Chang. He's a very kind man, so you can email him or send him some talk, so you can ask a question even after this symposium. Okay.
Thank you very much, Dr.
Thank
Kim.
you. Thank you,
So
Donglin.
nice to
Hope
see
to
you.
see you again sometime. Thank you very much. Now we would like to move to the second presenter. It's an oral presentation. So how an early flowering woody plant arranges its bud development from December to April by Day1, Kai-Feng Ma, Manzang, Xiyongzang, Zhaowang, and Qijiangzang. It's a Beijing Forestry University. So Dr. Day1 is ready. We're about 7 minutes early, but we can move to the next speaker. Okay, we have some internet problems, so we like to go with the video of his presentation, and then we can come back to the Zoom. Okay, video please.
Hello, 안녕하세요. My name is Yuan Xi from China. Very happy to attend this conference in Seoul. I'm a PhD student of Beijing Forest University, working on a Prunus mume project that led by Zhang Qixiang. My topic is how an early flowering cultivar of Prunus mume arranges its bud development in late winter. Prunus mume is a spring flower woody plant, belongs to Rosaceae, which include many other economic crops like apple, peach, apricot, almond, plum, and cherry, and of course, rosa. Among these, Chinese people deeply love Prunus mume, not only for the fruit, but also for the flower. Prunus mume could bloom under very low temperature, while others are still in dormancy. This blossom character of standing in severe cold and snow is seen as a symbol of unyielding and tenaciousness. Chinese people love Prunus mume, and particularly favor the first bloom one. So early flowering become our breeding target. Flowering is such a crucial event in plants' life cycle, for it's not only about ornamental volume, but also the yielding of fruit, seed, and petal. Different with herbs, flower development of many spring flower woody plants are segmented by endodormancy, ecodormancy, and dormancy release. Although many research had been conducted in crop, but they usually focus on endodormancy release or fruit set and nut harvest, leaving the flowering process remains unknown. How do plants sense the environment change to regular flower development? To breed early flowering cultivar, a series of work had been conducted in our team. First, we found one Prunus cultivar called 粉红朱砂, which flowering earlier than any other Prunus. Then we plan to carry out transcriptome analysis to dig the key gene of early flowering from 粉红朱砂. And for the transcriptome analysis to be more focused, a detailed investigation of bud development process should be conducted. So my report today is the first part of our work, the phenotype part. In this part, we would describe the bud development process of 5 different plants in late winter to early spring, including endodormancy breaking time, bud development speed, lowest temperature to bloom. We would compare those flowering characters for having a better understanding of the 粉红朱砂 flowering behavior, and then demonstrate the key reason why 粉红朱砂 can bloom earlier than other Prunus. Altogether, the result will be the basis to our next steps of work. Our breeding garden is located in the north of China, West Mountain area in Beijing. Material are all planted in the same hillside, grow with standard cultivate condition and without shading in the winter. The plant material, of course, our main character, 粉红朱砂, which bloom earlier in the field. Two other Prunus mume cultivar are selected from the field, called 早绿萼 and 美人. They are the second and the last blooming Prunus mume cultivar in the field. And for the better understanding of their flowering time, two other species of Prunus mume are chosen. They are Prunus davidiana and Prunus armeniaca variety, which bloom earlier than the peach and apricot that we are familiar with. Other detail need to be mentioned in the report is that in our experiment, every time we need to collect branches or buds data, branches from 3 sets of the plants would be chosen, as the picture shown, and the temperature data are collected by the recorder in the same area with experiment plants. And for statistical purpose, we define the development stage of flower buds as the picture shows. So when the first bud reaches stage 7, we will define that day as the first flowering day of the plants. So let's see the first flowering time. This is the first and the full flowering time of all experiment plants in the field. We can see that 粉红朱砂 is the earliest flowering one. 粉红朱砂 is 10 or 18 days earlier than the second one, Prunus davidiana, and about 1 month earlier than the latest one, 美人. Normally, endodormancy breaking time is the key factor of flowering time in temperate zone. Branches cut from all experiment plants are water planted in the incubator with proper temperature to grow. We record the bud break rate to evaluate the endodormancy is breaked or not. As we see from the table, the top 3 flower plants have more than half of the buds breaking the endodormancy in mid-December, and the Prunus armeniaca accomplished endodormancy in 1.5 months later. 美人 also break endodormancy in early December, but the last one to bloom. This result suggests that endodormancy breaking time is not the key factor of blooming order in northern China. Where we water planted the branches in incubator, it pretty much seems like this. It seems to have some correlation with blooming order, so we recorded how many days the buds need to develop from stage 1 to blooming of every buds on the branches we brought back. And the result is concluded below, that the top 3 blooming plants bloom earlier than the last 2. This result somehow explains the blooming order, but still cannot explain that 2 weeks gap of flowering time in the field. Then maybe the temperature to bloom is the reason. So we water planted the branches in the incubator with persistent temperature until all the branches flower or dead. Then repeat this process with different temperature to test the minimum blossom temperature of every plant. The result turns out that Prunus davidiana has the lowest blooming temperature, although not much difference between them. To summarize the above information, it can be seen that the top 3 blooming plants all have earlier endodormancy breaking date. All have earlier endodormancy breaking date, faster bud development, and lower blooming temperature. And it seems like that Prunus davidiana should be the first blooming one in the field. So how can 粉红朱砂 be the first blooming one among other prunus? Without an outstanding reason, 粉红朱砂 cannot bloom earlier that much than the second one, Prunus davidiana. Therefore, we conduct more detailed record in the field. Development stage of every buds on the 3 branches of each plant were recorded each time, and the percentage of buds in each development stage are calculated and shown in the bubble chart. As it's shown, we can easily find that 粉红朱砂 is 3 steps ahead of 2 other plants. And in the late February 2019, 粉红朱砂 already had about 6% bud reaches 6% buds reaches stage 6. They could bloom immediately when the proper temperature comes. And this is the key reason why 粉红朱砂 could bloom the first. Under the same temperature condition, 粉红朱砂 has a consistent effort under low temperature. So we wonder in which range of temperature. We conduct a gradient temperature increased experiment from 1 to 16 degree, 3 degree per step. The result shows that buds of Prunus davidiana has no obvious morphological change under 4 to 7 degree, while 粉红朱砂 keep developed under the same condition. This result consists with the temperature and bud data in the field from December to late January. Until now, we can conduct that 粉红朱砂 is the earliest blooming one in the field. It has many qualities to bloom earlier as others have. But the key reason for 粉红朱砂 to bloom first is the conduct bud development under very low temperature. Now our transcriptome analysis is on the way to go, and this result is surely can help us. Like if we want to dig the key genes of 粉红朱砂 early flowering issue, we would focus more on this part of the sample. And the following are some photos of our team. This is me counting buds. And my colleague who helps me a lot, 张志勇、马开峰、张曼、王佳 and 师傅王. My classmate and our team leader, my advisor 张启祥. He gives me many important ideas in the beginning of the research, and he's one of the motivation for me to getting on the research of flowering. Many thanks to them. And also thanks to the support program and our lab. Thanks for your attention. 감사합니다. 谢谢. The research of flowering. Many thanks to them. And also thanks to the support program and our lab. Thanks for your attention. 감사합니다. 谢谢.
Yes, we have some internet problem, so we turn on the YouTube for the audience. During that time, it was connected, and she started to speak through Zoom. So her presentation is a little bit behind our YouTube. So YouTube is finished. Now she's still talking. So Dr. Z, if you're listening to me, you can stop because every audience already watches YouTube, your presentation. So we can move to the question answer. So, okay, question answer. Any questions from the audience or viewer through the internet? Any questions? So, 프로노스 무메 is very popular in China, Korea, and also very, very popular early spring in Japan. Any questions? So, Prunus mume is one of the earliest flowering woody ornamental plants. So, she chose Prunus mume as her winter hardy and also early flowering characteristics. So, some pollination temperature is the lowest from the Fanhong Jusha. So, why they flower very early? That's the key point of the presentation. Any questions? We still have 5 minutes for the next speaker. No question? Oh, okay. Dr. Su, any comments from the speaker? Sorry for some problem between us. Go ahead.
Should I say something?
You cannot see the video.
Yeah, I can see the screen, but I cannot share my video.
Are you connected to the Zoom? No, it's a darkest. Yes, China is different, but it should be connected. But everybody can hear you, so you can answer to the question. But so far, no question from the audience or internet. Anybody? It's the first trial for the hybrid symposium.
I'm looking forward.
Very confusing.
Yeah.
Okay.
Yeah, sorry. Sorry for the internet.
Thank you, Dr. Z. So let's conclude your presentation. Thank you very much. Sorry for the inconvenience. Thank you.
Thank you.
Yes. Now we can move to the third presentation. The collection and breeding of hosta resources in Beijing Botanical Garden. At the beginning, as a keynote speech, we watch and listen to the hosta species in Korea, but this is from China. So Donghuan Liu, Mengmeng Zhang, Wen Qili, Xuwei Zhao, and Zue Qin Wang. Beijing Botanical Garden again. So, are you ready? Yes, please. Why don't you give him big hands? 이 소리였는데. 지금 당신 소리가 중으로 안 들린다. 그걸 좀 얘기해 주세요. Dr. Lee, can you listen to me? Can you hear me? In Zoom, we cannot hear you. So some technical problem. Could you switch on the audio? Could you try to turn on the audio, switch somewhere?
I'm sorry. I'm sorry.
Oh, yeah, now we can listen. So if you don't mind, could you
I go on.
go to the first and start it again? We have plenty of time, so if you start, it's 45, so we are
From
on time.
beginning or go on?
From the beginning, you can start again, if you don't mind.
Oh, beginning. Sorry.
Sorry for the inconvenience. Go to the first slide and you can study it over. Then
Sorry.
everybody can be happy. Now we can hear you very well.
I'm sorry. My name is Dongfang Liu from Beijing in China. At first, I congratulate the symposium. Then I thank to the committees to give me a chance to share my research results. My outdoor title, The Collection and Breeding of Hot Resources in Beijing Botanic Garden. There are 5 contents in my outdoor report. The first part, I give a brief introduction of Beijing Botanic Garden. Beijing Botanic Garden is located in Beijing, capital of China. Its latitude 116.28 degrees east, latitude 40 degrees north, latitude 61.6 to 584.6 meters. Annual average temperature is 12.6 centigrade. Precipitation 526.5 millimeters. High temperature is 37.7 centigrade. Lowest temperature is 17.5 centigrade. Their opening area 200 hectares with 1,000 texture preserved. It is the largest botanic garden in north of China. Among of them, ornamental crab apple, ornamental peach, rose, and horta are the important collected resources. It's a good place for plant research and public education. The second part, I introduced my research background. As we know, horta is what the famous perennial ground cover belongs to LHA. There are 3 subgenera, 10 sections, and 43 species, mainly distributed in China, Korea, and Japan. So long-term horta breeding, international registered cultivars reach up to over 6,000, enriching the plant diversity and the forests. However, there are only 4 species in China: Horta plantadenia, Horta vandicosa, Horta farinosa, Horta insata, which is restricted Horta application in China. This is my purpose for Horta research. The third part, I introduced collection of Horta in Beijing Botanic Garden. Horta was introduced in Beijing Botanic Garden since 2000 year from Holland, Belgium, America. There are 516 Horta resources preserved. According to leaf size standard, of a medical horticulture society. We did statistics for our collected hot resources. Most of collected are medium and small. They are very good ground cover. Then miniature are used to be potted. Only figure cultivars are giant and large. They are the very good background. Among there's 16 collected cultural species and varieties. It's a Hotter Plantagenesia, native to China. It has a large fragrant flower. It played an important role to Western Hotter breeding. It's a variety of Hotter Plantagenesia. It has double flower. It's a Vanticosa, native to China and Northern Korea. It has only bell flower, typically. It's a Hotter insata, native to China. It has typical patch rhythm, green division, and strong resistance. It's a Hotter latifolia, native to Japan. It's a first Hotter introduced to Western botany, the grandfather of Hotter ground cover. It's a Hotter sea body army, native to Japan. It has large and round blue leaves with typical wax powder. It's a Hotter kikuti, native to Japan. It has clear veins, red pink and dense flower flowering later. It's a Hotter ingari, native to Korea. It has typical spider shape flower, a good parent of breeding spider shape flower. It's a Tokudama, native to Japan. It has typical dome type with compact green fast, a very good ground cover. It's a Hotter Montana, native to Japan. It has strange type with large leaves and clear veins, early blooming. It's a Hotter nigrescens, native to Japan. It has high and strange plant type, a very good background. It's a Hotter fluctuans, native to Japan. It has typical fluctuant leaves, a parent of breeding fluctuant leaves. It's a Hotter pitching phyla and Equinoti asha, native to Japan. They have grape-shaped plant type, pea leaves. It has a dense later. It's a Kiyosumi incis, native to Japan. A very good ground cover. It's a Picoloca, native to Japan, with typical white powder at the back of leaf, a good parent of several breeding lines. Then I introduced some special cultivars. It's a giant leaf type, UFO. It has a giant leaf, leaf thick like disc shape, tree blade, resisting to sun and smell, a good background. It's impressed wool. It has giant leaf. It has deeply impressed veins. A good background on the forests. It's a Viking shape. It has giant leaf. It has a variety of the ground, sound resistant. It's a eclipse. It has giant leaf, thick and glacial green leaf with golden edge, strong resistant to sun. It's a final submission. It has giant leaf. It's tree blade. It has thick leaf with wide golden center, strong resistant to sun. Then Some is the largest leaf type, a guardian angle. It has a large blue leaf with a white center. Very similar to snail. A very good landscaping ground. It's Big Daddy. It's a largest blue-leaved cultivar cultivar in landscaping green in China. It's a sharp and sharp sense. It's the largest yellow-leaved cultivar in landscaping green in China. It's a strong resistance. It's a tetradyte glory. It's a tetrabloid, strong resistance to sun. It's a stained glass. It has a brilliant shine golden foliage. Have fragrant flower, sun resistant. It's a seducer. The golden edge is stable in 3 seasons. A very good hot cover. It's a good niche gracious. The golden edge is stable. A very good hot cultivar. There are 35 miniature hot cultivars. They are very lovely, used to be potted. Then, medium small leaf type of cultivars are very good ground cover. These blue leaf type, collecting blue azure, lilac. They are very strong, resistant to sun. Yellow leaf type, faint green tomatoes. It has a fragrant flower. It all goes small, a strong resistance to sun. It's a golden drop, very lovely small leaf type. It has strong division, 35% seed. It's a golden cedar with famous hot cultivar. Adapted to be cutted 35% seed. It's a golden center cultivar, the jewel, a lovely small leaf type with golden center, strong resistant to sun. It's a strong division. It's a white edge, white center, not before Christmas. It's a mango. They are cutted. They used to be cutted in 50% seed or 35% seed. It's a golden edge cultivar, Rego splendor, Academy blushing recluse. It's a knockout grand terror, very good grand cover. It's a white edge cultivar, Midman and Cedar Fair. Anno and Fancy. They are strong, resistant to sun and snow. They are collected tetraploid about 10. They are very strong resistance. These hot resources are broadly applied in landscaping green in China. Hot plant genia, Big Daddy, so sweet, grand flower, and flowering margarita. This hot cultivar and forest. Is a practicing as of forest. The 4 part update, I introduced my breeding of hota. According to the statistics of international cultivar registration of hota, there are 6 breeding methods. Most have no parent, covered 36%. Then spot breeding covered 28%. Cross breeding covered 17%. 17%. Hybrid breeding, self-breeding, seedling breeding covered less than 10%. Our breeding projects in breeding garden, whether petal or scape, dense flowers surround resistance. This is my first new cultivar through cross breeding. Female parent patron, male parent honeybell is offspring ready stout, has red scape and fragrant flower, ready treated in 2018 year. It's a second new cultivar breeding. A title Blood is a female parent, hot West Coast, native to China. Male parent essence of summer, it has large and round leaves, fragrant flower. Is crossbreeding. Is a generation 1 young plants. This their flowering period. We selected 3 new cultivar and registered in 2019 year. They are they have erect scape and grander and large fragrant flower. The first has a bell-shaped flower. 2 and 3 has tubular flower shape. They have strong resistance to sun. Then we have a spot breeding. This is spot of flowering magnetar. It's a floating milliliter. It's a tissue spot. They have strong resistance. Then it's a seedling selection, August Beauty. We selected 2 new cultivar, 舞女 and 白玉舞女 have word inflorescence. 白玉 has this flower. They are registered in 2018 year. It's a seedling of Volcano Island. It's a tetraploid. It has a red scape and good leaf substance.
Could you finish up behind the time?
It's a flower pot. As introduced my prospect. Breeding of color leaves the hot cultivars. Breeding of sun resistant foliar breeding. Thank you very much.
Okay, thank you very much, Dr. Liu. Sorry for the inconvenience at the beginning. We are about 5 minutes behind the schedule, but if you have a question, just one question from floor or internet. Any questions? Yeah, there is one. Dr. Lee, yes.
Thank you for your great presentation. My question is, have you ever...
Just a minute. Can you hear it? Dr.
Can you
Lee?
hear me? Do you hear me?
Yes.
Okay.
Okay.
Have you ever tried to mutation breeding such as gamma ray radiation or chemical treatment for your breeding program?
Mutation breeding.
I'm sorry. I'm sorry. Pardon?
Mutation breeding by gamma ray radiation or chemical treatment. for example, colchicine.
Irradiation. Okay. I think it's better just to move to the coffee break. So Dr. Seung-Yun Lee, you can contact Dr. Liu for detailed question.
Thank you.
Okay. Sorry and thank you, Dr. Liu. Let's conclude the first part of morning session, and we will have about 10 minutes coffee break. Thank you, Liu, again. Now we have 10 minutes coffee break. Thank you. Okay, now we move to the second part of first morning session. The title is Growth and Flowering Characteristics of Veronica, Native to Korea and Their Crossings. From Dr. Chun Yong Song, Sang Yong Kim, Cha Young Moon of Korea National College of Agriculture and Fisheries, and also Korea National Arboretum. Dr. Song, ready? Go ahead.
Okay.
Let's give him a big hand.
Thank you, Chairman. As you mentioned, I work Korea National College of Agriculture and Fisheries. Glad to meet you. Is that?
빼주세요.
Yeah. Okay. Sorry. The topic I present growth and flowering characteristics of Korean native Veronica and their some crossings. And next, the content like this, and then, do you know Veronica? Yeah, Veronica is classified as Scroflariaceae. Very difficult for me, the pronunciation. And English name, speed well, maybe grow well, speedily. And bird's eye, do you know Korean name? Maybe nobody knows the foreigner. The Korean name is 꼬리풀. 꼬리 means tail, priest friend. So the spike, what's the problem? The spike seems to like the tail of animal, like a dog or a cat. It's a very colorful flower, blue, like that, and the white. The point is problem. I can't. Anyway, no problem. And the color, rose, purple. And if you plant in the garden, the Veronica, you can see the flower spring to summer every year, because it's a perennial plant. Like that, the plant long flowering period, above 1 month, and easy to grow. So beautiful like this. And it's useful for the garden or pot plant, sometimes cut flower. Next. Actually, the Veronica naturally grow mostly in the northern fourth season. That means they. And next, like this, the plant, the Veronica is actually the green plant vernalization. So requires about 5 Celsius for 2 months. Then young plant, maybe out of battery. Yeah. When young plant for flowering, is a general process of Veronica growing in Korea. And next. There are many kinds of native Veronica in our country, around 11. Among them, 6 kinds of Veronica we are studied in this experiment. The Veronica Giussiana gravifolia in Korean, 큰산 꼬리풀, and Veronica Giussiana diamanica, 복내 꼬리풀, and Veronica dafurica, 구화 꼬리풀, and Veronica busanensis, and last one, Veronica filicillina. Like that, 6 kinds of Veronica were studied. Next. And their seed and data of native habitat supplied provided from the Korea National Arboretum, thanks to the Korea National Arboretum. And next, the data about the data of growth and flowering. The plant height was from the medium surface to the top of the plant, and the number of leaves larger than the 1 centimeter. Leaf length was measured the middle of the spike. And next, about the next, please. I don't have enough time, so quick. About flowering, number of flower is a total number of corolla in a spike. Flowering time from the day of the first one flowering to the day of falling down, 90% of corolla. The corolla color checked using the British color chart. Next, about the harvest seed. Actually, the number of harvest seed per spike or per capsule, like that, collected about 80 days after pollination. Okay, next. This is cultivation process of the Veronica. Seed sowing and germination September 1st in the greenhouse. After first transplanting, after 4 weeks of sowing, and then final transplanting November 25th. At the flowering time, we collected data of growth and flowering characteristics. And next is some complicated pictures. It's a crossing of Veronica, but we tried the full diagonal cross by 6 kinds of natives. So, crossing fully each other like that. So 36 combination, including the self-cross. And next. And the process of pollination, seed harvest of Veronica. First, castration of material line, the first picture, and then pollen collected from the paternal line. And then pollination by small brush. And next process is ripening seed. And 2 or 3 months after pollination, seed harvest like that. And next. Actually, the Veronica in Korea, natives on mountainous area, about 200 meter to the 1,200 meter. Meanwhile, the Veronica Giussiana diamanthus, 봉내꼬리풀, natives on the highest mountain in our country, the Sorak. The another one, the Busanensis, grow at the Busan city, the southeast big city in our country. Flat land. And then, next slide. Okay, no, before. It's a Veronica growth and flowering in the native or cultivation. First of all, the Veronica Giussiana gravifolia, 큰산 꼬리풀, distributed most mountain over South Korea, plant height above 6 centimeter, and leaf shape lanceolate of sedation. The raceme spike about 25 centimeter flower time, July to August, color color is like this, sky blue. And then by cultivation, next slide. It's by cultivation, plant height 75 centimeter, number of leaves 50 full around the leaf color, beautifully, yellow green. And the corolla color, purple. Spike length 27, and the number of spike 4 around the flowering time, June 13th to August. Like that, the growth condition is good for the Veronica in the greenhouse, so that the plant height and spike length much more than the wild one. Okay? It's natural. Next. Next one is Veronica piricillina. Natives 경상북도, that area. And the plant height around 40, leaf shape oval, completely split. Raceme spike about 15 centimeter, flowering on August. Color is bluish purple. By cultivation, next. Please. And the plant height 41 centimeter, number of leaves larger, medium, 60 around, the leaf color green, and corolla color light purple. Spike length 17 centimeter, number of spike 10, 11. Flowering time June 5 to August 13th. And next. About Veronica tahurica. This plant, native is most part of our country, about 50 centimeter. The leaf shaped oval, and the plant raceme, the raceme spike about 50 centimeter, flowering from August to September, summertime. The color is sky blue. And next. By cultivation, the plant height 51 centimeter, number of leaves 65. Leaf color green, corolla color also purple. Spike length 17 centimeter, number of spike 10 around, flowering in the hot summer day. Next. And next one, the 복례꼬리풀, Veronica giussiana, diamannica. Distributed sorak, the highest mountain in our country, above 1,700 meters, about 30 centimeter plant length, and the leaf shape of 8, flowering, the raceme spike, about 10 centimeter, flowering from July to August. Color is light violet. By cultivation, next slide. And the plant height 32 centimeter, number of leaves 40 around, corolla color purple, spike length 14, and the number of spike 9 around, flowering early to late summer. Okay, next. And next one, Veronica busanensis, located, grow the Busan, as I mentioned, around 20 centimeter height, leaf shaped ovate, flower raceme spike from the 10 centimeter, flowering from July to August. Color is blue. The plant shape is different. The postrate spread type. Okay, next. Plant height 30. Number of leaves 44, and corolla color also some dark purple. Spike length 13, number of spike 13 around, flowering time during summer, like that. Okay. And next, the flower shape and the plant shape is very unique compared to the others. It's the name of the Veronica nakayanum, 섬꼬리풀. As I mentioned, the grow at the Ulrum Eastern Island, around 30 centimeter plant height, leaf-shaped ovate, flower raceme, spike about 15 centimeter, flowering from June to July, early summer. Corolla color is light blue. And next, by cultivation, the plant height 35 centimeter, number of leaves 44, and corolla color unique, light blue. Spike length 15 centimeter, number of spike 6 around, flowering from late autumn to summer. Okay. And next, about the data, seed setting, seed harvest by self-crossing. the self-crossing, the number of seed of self-crossing per spike ranged 90 to 140, like that, or per capsule, 4 to 7, like that. The native Veronica tend to be fruitful to make seed compared to the other native plant, I think. Among them, the Geoshana gravifolia was the best formation of seed. As I mentioned at the material method, I tried the full diagonal cross each other, everyone. So 30 combination. However, the only 5 combination were harvested the seed by only 3 species, like that. 1, 2, 3, 4, 5. It's not easy to make seed by, I think, the kind of specific hybrid to make hard seed. Next, number of seed, their seed per spike around 40, around 30. It depends on some different space to space, but similar yield of seed, like that. Per capsule, 2 or 30. Okay, next. Yeah, from now on, very interesting, the result. There are 5 combination. The material line, Geoshana gravifolia, cross Veronica pericillina, the F1 hybrid, like that. How about this one? Beautiful, right? The phenotypic character, trait of plant or spike. How about similar to the material line, much more than the pattern line. In general, half in half, but this combination, some special, beautiful. In detail, next, the plant height 72 centimeter, number of leaves 88, leaf color, green. The material line is yellow-green, but green, this one. Corolla color, beautiful, purple. Spike length, 22 centimeter, number of spike, around 7. And the flowering time, June 18th to August 25, like that. This plant, the shape and the spike shape, beautiful. And flowering above 70 days, 70 days. So, I think it's very useful for the garden or pot plant, I think. Next combination is Veronica busanensis for material line and the pattern line pericillina, like this, the F1 hybrid. I think the plant type, some like prostrate, spread, followed the material line. So I think it's very useful for the garden, ground cover. Do you think? No other answer. Okay, next. In detail, the plant height 36 centimeter, and the number of leaves, many than others, 100 above, leaf color green, like that. Color purple, and spike length 10 around, number of spike 15, and flowering time during hot summer day. Okay, during hot summer day, there are few flowering plant. Okay, next. This is a reciprocal combination. The first one, the material line, the Veronica pericillina, pattern line, Geoshana gravifolia, 큰산고리풀. Seems like, took after the material line and pattern line in a half and half, I think. And next slide. I don't have enough time. Yeah, so many slides in there. The plant height 60, and number of leaves 114, and corolla color purple, spike length 15, around the number of spike 11, and the flowering time June 17th to August 21, summer day. And next combination, Veronica Piricillina, cross Butanensis, the F1 hybrid. I think it also follow the maternal or paternal line, half in half. And next, in detail, the plant height, 52 centimeter, number of leaves, 128, many than others. Color light purple, and the spike length, 12, number of spike, many than others, 13. And the flowering, June 15th to August 27th, the hot summer day. Okay, and the last one, last combination, Veronica butanensis cross the Guiana gravifolia. It also, the F1 followed the maternal or paternal line, the half and half. Okay? Next, plant height 41 centimeter, number of leaves 126, leaf color green, beautiful, and corolla color purple, spike length 13, around the number of spike also 13 around, and flowering is summer day. Okay. Do I have time to say the conclusion? Okay, thank you. Okay, time to conclusion. Yeah. South Korea natives approximately 11 kinds of Veronica as a perennial plant, which are natives mostly mountainous area from 200 to 1200 meters. When the native Veronica was grown in the greenhouse, the growth of plant height, number of leaf or spike length was increased than that of inhabited native itself. The number of ripened seed per spike was different, depend on species, ranged from 90 to 140. In the crossing of natives, the growth and flowering characteristics, such as the number of leaves, spike, were increased compared to the native itself. For example, the crossing of Veronica Guiana gravifolia cross Piricillina had a plant height about 70 centimeters and long flowering above 70 days, as I mentioned. So I think it's very useful for the garden plant. Okay, that's all.
Thank you, Dr. Song, especially for finishing on time. I should have mentioned that this was an invited speech, so I gave him 25 minutes. So do we have any questions on his presentation? I have worked on the Veronica with 2 species, and I saw many Veronica, the cultivars in the United States, but it's amazing, amazing. I could see the heterosis, the last one, so much bigger by crossing. It's great. Any questions?
Thank you. I talked puppy, so.
In the internet, any questions? Then let me, yeah, there is one. Excuse me, just a minute. Microphone, so that everybody can hear you.
Yeah, thank you for your presentation. So myself, Sivanation from Gonguk University. I would like to know, did you find any difficulties in germinating seeds from the hybrids? And in case of inducing flowering, did you find any temperature differences? for inducing flowering for the new hybrid?
I don't understand perfectly what your question. What's the question?
Seed
First
germination?
question is like seed germination.
Germination?
Yeah.
Yeah, proper germination temperature about from 15 to 25 Celsius degree.
Any issues, any problem in
Yeah, any species at that temperature, no problem to germination.
Okay, the hybrids all germinate well, right? I'm talking about your hybrids.
Hybrid also.
Yeah, okay. So my next question is any variation in the flowering temperature? Did you find out?
Variation.
Variation. Yeah. As I mentioned, from material to partial line between
Okay, so
that the hybrid.
Day temperature is 15, night temperature is 5. When you increase the night temperature above 5, like a 10s, the flower, it will flower? That's my question.
뭐라 하나 잘 모르겠네. 통역 좀 해주세요.
Thank you very much. How do you convince that you made your F1 hybrid that is actually true interspecific hybrid or not? 어떻게 그 F1 잡종이 확실히 종간 잡종인지 아닌지를 어떻게 확신하느냐는 걸.
네, just very good question about my presentation. The F1 hybrid follow up. Sometimes the motor or petrol line, half and half. So I can find out the phenotypic growth, the plant or spike.
Yeah, I think so. Thank you very much. Yeah.
You understand?
Yeah. Morphologically, you mean that morphologically in between from the characteristic of parent, right?
That's right.
Yeah.
Yeah. Phenotypic trait.
Yeah. Thank you.
Okay, the translate.
Let me
Thank
have
you.
one question.
Huh?
Yeah, since there is no question. Okay, as you mentioned about the ground cover by Veronica. As ground cover, you said that it's a spreading type.
Yeah.
In the woodland or garden, there's no problem. But in urban area, we have many de-icing salt, or some seashore area, a lot of the sea salt problem. So, have you tried find out any salt tolerance of Veronica species?
Okay, as I mentioned, there's some combination proper useful for the garden, the ground cover.
Yes.
Yeah, some water line like Veronica Giussiana diamanica spread prostrate.
Yeah.
If I use the line, the F1 follow up the
내염성이 어떤지, 내염성이 있는지. Salt tolerance. What about the salt tolerance?
I didn't check the. I didn't check.
I think we need to test the salt tolerance to use in the urban areas.
However, the Veronica is make a beautiful garden during hot summer day. During summer there are few plants to flower in the garden, but the Veronica is work.
Thank you very much. Let's conclude first invited speech as a second session. Thank you.
Thank you. Thank you for your attention. Yeah.
Next, we'll bring Dr. Donglin Zhang from the University of Georgia again. He's going to talk about the genetic inheritance of hydrangea macrophylla clones using ISSR markers. As I mentioned before, he's from University of Georgia, and the co-author is Jian Song. He's a Flower Research Institute of Guangxi Academy of Agricultural Science, China. Donglin Chang, can you hear me?
Dr. Kim.
Okay.
Appreciate it. And well, nice to see you again and talk to you about the genetic relatedness of hydrangea macrophylla clones use ISSR markers. And I would like to share with you the work. Actually, I did not do anything. And Ms. Sun Qian did all the slides and the work, and with the help of Dr. He Li, and both of them, they contribute to this project. I'm just an advisor. So today I would like to present on behalf of them about hydrangea. Everybody probably knows hydrangea is a beautiful plant. There's so many hydrangea in this world, right? That can use for cauliflower, can be potted plants, also can use as dry flower. Definitely the garden. Seems like people said, if you don't have hydrangea in the garden, it is not a garden. So they really have great potential. Some of you maybe heard recently they gained popularity because of reblooming structures. So this is probably something I want to mention a little bit more about you, because this is what happened to track this project. So what we do is we have a question was posed. Say, if you have a genetic, the morphologically similar characters, similar plants, how do you know the difference or not? A lot of words, you probably know Endless Summer is originally from my lab, and is really popular and gained so much popularity because of those reblooming. Now we have several other clones, very similar, right? Include one of the parents, Goliath, is also from those Endless Summer parents. So there's so many and new clones of Endless Summer, and also the old one. How do you distinguish, right? Morphologically, they have a little different, but a lot too much. So what's this question? We try to use ISSR markers, quickly answer this question. Please be advised, this is a lot of thoroughly study. This is just a preliminary screening study. And don't think about the work is a lot complete. The work is just give you preliminary ideas and how we can verify the clones are new or not. So what we have is, in our position, we have 9 related hydrangea, technically 8 related. 1 is from Japan, and the other they related. 1 is from Japan, is a little bit far away. What we try to do is, we try to take the DNA and do the ISSR markers, is try to answer whether they have similar or not. As you see here, this is a clone, except the one clone, the ISO and this one is a double flower from Japan. All others are more or less related to Endless Summer. So this is probably one question I want to, for example, bishop is kind of a mutation and have a large mop head inflorescence, sterile flower, and pink to blue. Those kind of plants really hard to distinguish with the others, right? Bloom strike is a new cultivar from Endless Summer because of stronger stem, they can handle the garden solution better. And also we have a traditional Endless Summer. This was released back a few years ago. We get a lot of royalty from those plants because they're so popular. Goliath is another cultivar with a kind of a serrated tipos, but this is an old cultivar. Do join those breeding for the Endless Summer. We know that. And also green mantos is the tipos come to green which is really, really different than it's a mutation from this glias in terms of morphology because the color different, right? And this is why it's only different. We use those as comparison because ISGO2020 is from Japan and double flower majorly is a lace cup, right? So this is probably we want. Marcus gold is a variation of golden foliage, which I don't know how much difference. And Trace and Xiao is another endless summer series. Majorly is lace cup flower. For some reason, they have one plant at the UConn campus. We take cuttings. They have both lace cup and mop head on some plants. So it's kind of very strange about those. Tristan Xiao is a kind of rebloomer, very, very popular and better than endless summer, but different inflorescence. So this is a kind of clone I want to introduce. Technically, again, 8 of them is more related and only ISGO2020 is a little different. So this is probably I want to share with you. What we did is we take the DNA and the extraction, and when I was a student, is take a publication to extract DNA. Now you can extract DNA from anything. So I don't want to get more details about those, just want to know you. So one part is you need to do is you need to trial, especially the temperature, right? Annealing temperature is very important. So when you do this PCR, it's pretty regular. You just try, make sure that what is the best for your amplification, right? When you do ISR, there's so many primer available. You can say there's so, and the UBC Columbia selected most popular 100. Actually, each SSR markers you can use as a ISSR markers. So in order to select the best primer for those, we use the 2 closely related Goliath and the green mantle because they are very related. And then we use a distance far away of ISGO2020. So that way we can figure out what the produce best band and show the similarity and the difference. As you say, UBC primer 815, you can say the very similar except this band, and then ISGO2020 is very different. So we selected 10 primers based on those distinguished, right? We definitely screen 100. This is the best way. Don't be lazy. It is very, very good if you do thoroughly investigate. Then what do? We amplified, use the 10 selected primer, amplified all line clones. Then you use to say the electrophoresis on the gel image. Here is a primer information we used. As you say, if you have clone 1 and clone 2 have a little difference band, you can say they are polymorphism. As you say here, the polymorphism is pretty high, even though they're closely related. The highest definitely we used the highest primer is 847. You have 90. The polymorphism is very high, right? 847. You can CAC. Now CA. Repeat. This is a probably don't be afraid or try to save in time, because if you don't do this well, you have to pay back later. So thoroughly invest about the primer selection. They use all the selected primer to amplify all 10 of them. This is the result. You can say again, each say, for example, UBC 8035 primer, you can compare the first 3 clones. You can share the band, right? You can say a little bit, then you can say a polymorphism band. So those band pattern is quite different. Now, after those, you have to read it by machine or by yourself. So both way we have to check. What we found? We found out the genetic similarity and the distances. Number one is if you look at those 8, which is Yukon, and Tristan Xiao, and then Tristan Xiao is very similar. There's not too much difference, very similar, probably individual difference, even though this one have little bit both ring flower, the lace cup and the map head, right? If you do the UGMA tree, the UPGMA trees by clustered label joining or the other joining method, you can see here the 2 plants we just mentioned almost identical, right? It's a low different. The only difference is the ISGO2020. They are very different compared with others. So all other clone almost identical because when the Anthony Summer was released, do use green mantle and the Goliath as a parent. They have a little different, but it's clustered as Anthony Summer. All other is a derival from Anthony Summer. So as you say here, we can distinguish a little bit, but not as thoroughly as we want. At least what we try to do is answer those one are different or not. So to keep this in mind, what we concluded, the ISSR primer, 10 primer selection of the ISSR can tell the difference of hydrangea clones, right? Not thoroughly, but it can. So if you think about the 2 types of flower, usually we think about the 2 types of flower are huge difference, mop head and lace cup. Technically, from those results, we don't say too much difference because one plant has both flowers and the DNA tell me. So double flower cauliflower ISGO2020 is definitely different than any others. So we conclude ISSR markers can be useful method to distinguish hydrangea clones and it can be used on morphological similar plants to distinguish. So like the question about the Veronica, right? How do you know it's a true hybrid or not? Now we can use DNA, can easily tell you. So this is what we want to share with you, and hopefully you get some information to solve the problem. Again, this is just a preliminary screening study, not a thorough study of genetics or some other method. Hopefully you can take a message and then do some identification for yourself. Thank you very much. Any questions?
We can set 1 question. Any questions? Yeah, Dr. Im Ki-byung, yes, give him microphone.
Thank you, Professor Jiang, for your valuable presentation. I just want to know that you already have done some other species for the discrimination of using ISSR marker. That's one question. And also, have you done any chromosome level research for the species of the chromosome cytogenetic study?
The first question is, yes, we tried the ISR for several other plants, such as Ilex cornuta. We have a student that did those. We did Ilex hydrangea, other species level 2, and we just don't have good data to publish because we don't have enough species. We also did Distilium, and they have publications on the ACTA too. This is many years ago. We did from other plants too. ISR did quite a bit. Flowering peach and all others, we did quite a bit of plants. And in terms of crimson study, hydrangea is kind of a weird plant because the crimson number is quite different than others. We have not done anything about the crimson study. So maybe you guys can do some work on it. We did not. We not intend to do that. Thank you very much.
Yeah, maybe we can collaborate each other.
Oh
for the
gosh, that would be
chromosome,
great. I
study.
really want to go. I have two chances to go to Korea this year. One is the Magnolia Society meeting. Now it's this meeting.
Yeah.
This is a COVID. Just stop everything. I just miss it. Look forward to visit you someday.
Okay,
Yeah.
let's keep in touch. Yeah,
Thank you very much.
Thank you.
Thank you again, Donglin.
Thank you.
Okay. Now we have to move to the next speech. Next one. Breeding of new ground cover chrysanthemum cultivars with the compact plant architecture and rotching resistance by Kong Chuan Yuan, Qi Jiang, Ming Sun, Tangren Cheng, Jia Wang, and Huitang Pan. So Dr. Yuan is ready.
Thank you, Chairman. Hi, friends from all over the world.
Can you hear me?
My name is.
Can
Hello?
you hear me, Dr. Yuan?
Yeah.
Okay. Go ahead.
Okay. Can you hear?
Please turn on the video so that we can see your face. Video.
And can you hear me? And
turn on the video, please. Dr. Yuan?
Yeah. I can hear you.
Turn on the video. We cannot see your face. If you don't mind, we'd like to see your face. Turn on the video.
Can see me with my face. Okay. Thank you, Chairman. Hi, friends from all over the world. My name is Cun Quanyuan from College of National Engineering Research Center for Floriculture, Beijing Forestry University. I'm honored to have the opportunity to speak at this conference. Today, I would like to share with you some of my research progress of breeding of new ground cover chrysanthemum cultivars with compact plant architecture and lodging resistance. Ground cover chrysanthemum is an important cultivar group in chrysanthemum. Due to strong cold and drought resistance ability, strong adaptability, extensive management, and dense flowering, the ground cover chrysanthemum was widely used in highways, squares, streets, parks, scenic areas, and residential landscaping. However, most of existing varieties are upright and have tall plant height. Owing to this, the plants are prone to fall down in full bloom period, especially after rainfall, and varieties with compact plant architecture and lodging resistance are rare, which limits its promotion and application in landscaping. How to solve this problem? The answer is to breed new ground cover chrysanthemum varieties with ideal plant type. The ideal plant type of ground cover chrysanthemum means the plant is short, creeping, and round. The branches are numerous and compact. The crown is large. The ability to cover the ground is strong. The color and pattern are unique and rich. The flowers are dense. The leaves are almost invariable during the full bloom period, and the plant has active lodging. To breed new chrysanthemum cultivar with ideal plant type, the wild chrysanthemum species of Chrysanthemum yantins with prostrate growth characteristics was used as an important parent. It is a rare new species of Asteris with super prostrate characteristics, multiple branches, strong ground cover ability, strong salt resistance, wide adaptability, and large numbers of flowers, which was discovered and named by our team from Yantai of Shandong Province, China. Through successive multiple generations of hybridization and selection, new varieties of ideal plant architecture were obtained. The outstanding features of new varieties are low and compact. The height is less than 45 centimeters. The plant is prostrate. The crown width is nearly 1 meter, and no lodging during the flowering period. The natural flowering period is from early September to late October. The cultivated new varieties have great potential to be used in garden and ground-covered applications with better landscape effect as a group application. The other type is the new varieties with, oh, I'm sorry. Sorry, is there some problem? I'm sorry. Sorry. The other type is new varieties with short plant height, lodging resistance, compacted and rounded type at the death flowering. The silent features of this type are compacted with strong branching ability and growth in a hemispherical shape without artificial pruning. Sorry.
Then make a full screen. Then it works.
Sorry. Sorry, have some problem. Sorry. I can't open my camera. I continue. The other type is a new varieties with short plant height, lodging resistance, compacted and rounded type, and death flowering. The silent fractures of this type are compacted with strong branching ability and growth in a hemispherical shape without artificial pruning. Densely flowered and hardly see green leaves. No lodging during flowering period. In addition, after years of breeding, the flower type or ground cover chrysanthemum has been greatly improved. New varieties with some attractive flower types were obtained, like petals with irregular curly type, and mosaicic color type. This greatly enriched and enhanced the ornamental and application value of ground cover chrysanthemum. Several varieties were exhibited and won several awards at the International Horticultural Exhibition 2019, Beijing, China. The new ground cover varieties have great potential to be used in community landscape for flower sea, highway, square, street, park-scenic area, and residential area. It is an excellent material for arranging group landscape. It can also be used for pot plugin. Create new germplasm ground cover chrysanthemum, the ideal plant type. Our laboratory has carried out continuous crossbreeding and selection work for many years. By introducing the genetic information of wild chrysanthemum species, we have obtained a series of new and super germplasms with prostrate growth characteristics, providing excellent materials for landscaping. However, the resistance varieties such as cold resistance, drought resistance, disease and pest resistance still needs further evolution. In addition, there is still room for further improvement in terms of flower cover, flower pattern, etc. In the future, we will continue to breed more new ground cover chrysanthemum varieties with ideal plant types, abundant flower types, and excellent comprehensive traits. At the same time, we are carrying out research on the mechanism of chrysanthemum branching and prostrate growth, so as to lay a theoretical foundation for the breeding of new chrysanthemum species with ideal plant type. Finally, I'll thank Fundamental Research Funds for the National Natural Science Foundation of China, National Science and Technology Program during the 12th five-year plan, and the Special Fund for Beijing Common Construction Project. Meanwhile, I want to thank our team members, especially Professor Qixiang Zhang, Dr. Taran Chen, and the students of Xiaowei Li, Yujie Yang, Juntong Chen, and Qinqin Yang. All of the above work is a team effort. Okay, this ends my presentation. Thank you for your attention.
Thank you very much, Dr. Yuan.
Thank you.
We have 1 or 2 minutes for the question. So is there any questions from the floor or the foreign country? Any questions? Dr. Yuan, can you hear me?
Yes.
Yes. I have one question. You are talking about the prostrate ground cover type chrysanthemum.
Yeah.
So since it's stick to the ground, with high humidity underneath, and is there any chance for some disease problem?
Resistance such as cold resistance, temperature, and disease resistance, such as cold resistance, temperature, and disease resistance and resistance. This is very important. And as the first step, we have evolution this resistance, such as cold resistance, but some disease resistance still needs further evolution. And in the future, we will work hard to evaluate this resistance. Thank you.
Okay, thank you very much. Now we move to the next, the last presentation this morning. Since we cannot contact her through the internet, we are going to show you the video. And after video, since we cannot make questions to her directly, so you can use the email to her. Okay, is the video ready? Chrysanthemum genome. An in-depth study must be performed Tuan Kang Nguyen, Suong Tuet Tiha, and Dr. Jin Hee Lim, Sejong University, Korea. Okay.
Hello, my name is Tuan Kang Nguyen from Sejong University. I would like to present about chrysanthemum genome. An in-depth study must be performed. I will talk about introduction. As we know, chrysanthemum species have numerous considerations of flower colors, types, and shapes to be widely used as an herbal or commercial product. Chrysanthemum plants have various polyploid hybridization from diploid to decaploid. Chrysanthemum genome has self-incompatibility. Heterozygosity is very high and difficult to establish a group of genetic research. It is very large and statistical report around 2.7 GB with 32.3% GC. Next generation sequencing was applied in chrysanthemum nankigensis in 2013 and genotyping by sequencing applied in wild chrysanthemum in this year, and long-read sequencing study on chrysanthemum moriflorium transcriptome reveal flavonoid biosynthesis and regulation. I will talk about genetic mapping and QTL study in chrysanthemum. Song et al. Study on high-density genetic map construction and identification of loci controlling flower type traits in chrysanthemum. As we know, flower type is an important and extremely complicated trait of chrysanthemum. Two case factors affecting chrysanthemum flower type: the correlative mass degree, we call CTMD, and the relative number of rainforests, we call RNRF. 305 F1 hybrids were obtained from two parents with obvious difference in CTMD and RNRF performance. For CTMD, they found three major QTL in LG1, LG3, and LG11. They also found 15 QTLs in LG4, LG6, LG14, and LG20 related to the number of rainforests. We can see the female parents and male parents and a selection of the hybrid offspring. I will talk about chrysanthemum GWAS study. Su et al. Study on GWAS and suggested the candidate gene for waterlogging tolerant in chrysanthemum. They used 92,811 of 88 chrysanthemum accessions, including 64 spray cards and 24 dispersed chrysanthemum. We can see the phylogenetic tree. I will talk about chrysanthemum transcriptome analysis. Dong et al. Study on transcriptome analysis of the molecular mechanism of chrysanthemum flower color change under short-day photoperiods. As we know, the flower color of chrysanthemum blossom induced by artificial short-day treatment were lighter than those induced by the natural photoperiods. Transcriptome analysis of chrysanthemum morifolium gene beta horn revealed that the biosynthesis of anthocyanin is tightly regulated by the photoperiods. We can see the highest level of anthocyanin accumulation appear in the fall blossom stage under the natural daylight condition. We can see figure 9, the heat map of anthocyanin structural gene expression level, 62 unigenes encoding 8 enzymes show light chain during the flower development. I will talk about chrysanthemum mRNA study. Yang et al. Study on UVB radiation responsive microRNA and their target genes in chrysanthemum using high throughput sequencing. They reported that several mRNAs related to flavonoid biosynthesis such as BC3P, 114, 893, 93, and MIR 4367P5 were inhibited by short-term UVB radiation. This suggests that the contribution of UVB to flavonoid accumulation depends on a low dose of UVB radiation. We can see figure 1, the effect of UVB radiation on bioactive accumulation of chrysanthemum. UVB 12 hours so high solute sugar and levoglucogenolic acids and laurogenic acids. I will talk about genome study of chrysanthemum. Hikarawa et al. Did the de novo whole genome assembly in chrysanthemum certicubis, a model species of chrysanthemum, and its application to genetics and gene discovery analysis. They use Illumina sequencing and reported that SMRS10 chrysanthemum certicubis accession developed by 5 generations of cell crossing from A cell compatible strains AEV2 were used for genome sequencing. Here is the phylogenetic tree of the phosphatidine ethanolamine binding protein, and phylogenetic analysis and amino acid alignment using depth from other plant species suggest that only 3 FT ligene we call CHFTL123 and floral promoter activity, while for TFL1 and CEN PFT ligene have floral repressive activity. CEN gene is highly expressed in the short tip of the rosette, forming dormant glands that are incompetent to initiate flowering. So they saw 2.72 gigabyte of assembled sequence and consists of 354,212 scaffolds, cover 89% of 3.06 gigabyte. Chrysanthemum city cubic genome estimated by k-mean analysis. In gene recovery analysis, candidate flowering related genes were newly found in CSE R1.1 CDS. Chrysanthemum city cubic genome were applicable to genetic analysis in cultivated chrysanthemum. I give the conclusion by the use of high technology such as QTL mapping, GWAS, transcriptome, and mRNA to apply in various development stages and environmental conditions. Chrysanthemum breeding will be performed and discover molecular markers through gene function research, and that can be used as a reference for genome decoding in chrysanthemum plants. Currently, the long-range first-generation equipment such as Pacific Bioscience single molecule real-time we call SMRT sequencing and long-read sequencing technology from Illumina sequencing platforms. It is expected to improve the integrity of the genome by reading and assembling sequencing longer than the length of the repeat. Thank you for your listening.
Thank you, Dr. Nguyen. As I mentioned before his presentation, he is not connected to our program at the moment, so we cannot ask a question. So you can, if you have a question, you can send him an email, so he can answer your question. So now we can conclude the morning session. Thank you your cooperation, and sorry for the inconvenience we had, especially at the beginning. Thank you very much.
Thank you. Thank you very much for your wonderful management of session 1, Dr. Kim, and all presenters. Before we have lunchtime, we will have group photo. So please come to the stage here, and for our online participants, please connect to the Zoom with video on and without any voice. So if you are still on our Zoom connection, yes, we can just show you on our video. Yes, now we are preparing. So for our YouTube or Darkcast participants, please wait a minute, then you can maybe share for our group photo timing. Could you just come into the stage?
Everybody, please come forward for the group photo.
And we can see very well for our Zoom participants. Thank you for all cooperation. So please keep in touch with Zoom. And actually we have so many people in Zoom, so maybe we can move on the slide number and we will take some pictures, like 3 or 4 cut.
Could you bring some chairs? Chairs. Huh? Maybe about 10 of them for the first line. 의자 좀. 저 어르신들 그 가운데 앉으시고. 오케이.
Professor Lee, can you control the Zoom screen with more people together? There are just 9 people here, so can you just modify the screen with more people together? Thanks, Dr. Zhang and Ryan and Yuan and everybody. We can see you very well, so please keep in connect on Zoom. And we will take a photo maybe 3 or 4 times because there are some limited people on the screen. Okay, we are almost ready here at the venue, and we can check your face on Zoom participant. Maybe just 1 minute. 네. Yes, we are almost ready. Maybe just 30 seconds. We will have one more photo for our participants via Zoom. And Dr. Zhang and Ryan, don't worry about your face. Maybe we can just figuring out that we can see you very well. So through the monitor, YouTube, your face is not on for full shot, but we can figuring out here from here. So don't worry about our good photo shot. stay on Zoom, we are now modifying the light. More darker condition is better for the photo. So we are now adjust to the light condition. 아니 저 이거 라이트 끄고 다 같이 한번 찍고. 라이트 지금 끄고 계신 거 같아요. Please stay on Zoom, participants. We would like to take your photo only, except for the participants at venue. We are taking a photo for our Zoom participants. We appreciate your cooperation. So for all of you from Zoom, now we are trying to figuring out the best angle. So could you just stay for 1 or 2 minutes? Yes, our organizing committee is taking a photo for our Zoom participants now. 됐어요? Everything is good? 됐을까요, 위원장님? All right, thank you so much for everyone. I think this is very unique experience for all of us. 화면 저 비춰주실래요? Okay, so now we have lunch time, and for online participants, we are preparing the photo exhibition and video clip by Korea National Arboretum during our lunchtime. And please enjoy our Korean folk plants during your own lunchtime. And I would like to have you at 1:30 for session 2 by Chair Chun Young-song and Wan-Sung Kim. And there will be our invited presentation by Dr. Ryan from Oregon State University. So please come and join us at 1:30 again. I will see you soon. Thank you.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean peninsula. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. Let's meet the preservation team, who are working to protect Korea's endemic plants. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. The forest is an open-air learning site of life. They learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum.
Where the present and past coexist. Korea National Arboretum. While everyone is busy chasing today, the forest is changing at a slow pace. A secretive 550 years. The start of which can be found at the Royal Forest in Gwangnam, where the tomb of King Sejong, the 7th king of the Joseon dynasty, and Queen Jeonghye are located. The Royal Forest has embraced the tomb over time. From the nation's experimental forest dedicated to research and study of forestry, to an arboretum researching vegetation and forest today. The forest changes every day, telling us many different things. Meeting diverse plant life over 4 seasons. Studying the bountiful gifts of nature. A glimpse into the precious life of nature's monuments and hidden existence of rare species can be found here. The forest has become fertile and its value heightened. Designated by UNESCO in 2010 as a biosphere reserve, the nation's largest experimental forest research center. The nation's only research institute dedicated to the diverse species of the forest. Our nation's representative forest ecosystem report. It has become a resting haven for citizens, where the environment and culture harmonize as one. Think about the tomorrow that the forest brings us. A forest of healing that we seek out in times of need. A forest of hope to find the sounds of nature. A forest to bring us back to the beginning, to cherish nature's natural beauty. a treasured forest for our tomorrow. A space which is shared by the present and the past. Dreaming of a beautiful tomorrow, living in coexistence. We look forward to making a 1,000-year history at Korea National Arboretum.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. Gwangnung Forest, where the Korea National Arboretum is located, has been protected as a royal forest since 540 years ago. Possessing a stunning natural environment, it is the habitat of life, ranging from rare plants to natural monuments. It's why the value of Gwangnung Forest was recognized globally and designated as a UNESCO biosphere. Gwangnung Forest is a natural masterpiece with a harmony of life, presenting unique and stunning scenery in each of the 4 seasons. It's a new morning at the Arboretum. The Korea National Arboretum is a special place where you can see the Gwangnung Forest and the various organisms that inhabit it. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. Today, it studies plants and insects that inhabit the forests, researches seeds and specimens, organizes exhibits, and provides forestry education. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The specialized gardens include the Ornamental Tree Garden, presenting a collection of trees with high ornamental value. The Flowering Tree Garden, which exhibits trees with beautiful flowers. And the Aquatic Plant Garden, Medicinal Plant Garden, Rare and Endemic Plant Archive, and Temperate House. The Korea National Arboretum also consists of specialized facilities for specialized activities. The The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It contributes the collection and preservation of tropical plant resources of infinite value, and to secure future resources which will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Today, the team is here to conduct an extensive study of the organisms that inhabit the Paektu Mountains. The researchers observe each flower and tree, recording their number and characteristics. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. The efforts to protect the beautiful natural environment and biodiversity must continue until we complete the biological survey of the Korean Peninsula. Let's meet the preservation team, who are working to protect Korea's endemic plants. The Korea National Arboretum is leading efforts to preserve life forms throughout Korea. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Recently, the Korea National Arboretum successfully revived a rare orchid through proliferation. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Throughout the country, there are restoration sites that have been restored by the Korea National Arboretum. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Forest organisms collected throughout the country are brought to the Korea National Arboretum. Creating specimens that preserve the natural forms of forests and studying the insect ecology are crucial steps in securing important biological resources. The Korea National Arboretum is recognized as the base camp of Korea's biological resources for its systematic management of specimens and plant seeds. Through this effort, the Korea National Arboretum builds a world-class biological resources knowledge base of organisms in Korea. Preserving plants that represent each region is one of the Korea National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Discovering the potentialities of plant resources is crucial for our future. Proliferating endangered plants, restoring and improving them, the Korea National Arboretum is working to unleash the full potential value of the plants. It is also discovering the infinite power of the plant genes. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korea National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. The Gwangnam Forest is a UNESCO biosphere, which means that its value is recognized by the world. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korea National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korea National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. Today, we have delightful guests at the Korea National Arboretum. The Forest Museum is a popular learning site as you can see all the plants of Gwangnam Forest in one place. It is considered the best place to learn about and to understand the forest. The forest is an open-air learning site of life. As visitors touch and experience the forest hands-on, they learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The hands-on experience in the forest offers learning for everyone, regardless of age. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum. but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum. Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are protecting the lives in the forests. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. Let's meet the preservation team, who are working to protect Korea's endemic plants. Observing the habitat of plants and protecting endangered species are two of the Korean National Arboretum's key roles. Reviving endangered plants is also very important. Each year, the Korean National Arboretum designates endangered rare plants and restores them in a systematic manner. Since we can't bring back nature once it's lost, the Korean National Arboretum's efforts to protect this nature must continue. Preserving plants that represent each region is one of the Korean National Arboretum's important roles. Preservation of such plants is crucial in determining and researching the habitat and environment of Korea's endemic and rare plants. Proliferating endangered plants, restoring and improving them, the Korean National Arboretum is working to unleash the full potential value of the plants. By forming the DMZ National Arboretum to preserve the forest ecosystem of the DMZ, the Korean National Arboretum is securing the biodiversity of North Korea's plants and is studying rare, endemic, and endangered species. Through systematic preservation activities and exchanges with arboretums and botanical gardens overseas, the Korean National Arboretum is leading efforts to study forest organisms around the world and discovering their value. The Korean National Arboretum is hosting a daily gardening competition to expand the gardening culture in everyday life and supporting the development of gardens in schools using Korean wildflowers. Furthermore, it is working to increase global awareness of Korean gardens. The forest is an open-air learning site of life. They learn about the various organisms that inhabit the forest, as well as interesting tales about the forest. The BioBlitz, a biodiversity exploration competition, is a great opportunity to learn about all the organisms in the forest, including plants, animals, insects, and reptiles. It is a time when people can interact with the forest and the organisms in it. After the hustle and bustle, the day is over at the Arboretum, but the meaningful efforts of the Korea National Arboretum will continue throughout the year. Korea's leading forest organism research institute, Korea National Arboretum. Based on its outstanding achievements, it is now preparing to soar to new heights. By improving its collection, research and preservation of forest resources, and expanding the scope of forest research, Korea National Arboretum will establish itself as a world-class arboretum. Korea National Arboretum.
Where the present and past coexist. Korea National Arboretum. While everyone is busy chasing today, the forest is changing at a slow pace. A secretive 550 years. The start of which can be found at the Royal Forest in Gwangnam, where the tomb of King Sejong, the 7th king of the Joseon dynasty, and Queen Chunhee are located. The Royal Forest has embraced the tomb over time. From the nation's experimental forest dedicated to research and study of forestry, to an arboretum researching vegetation and forests today. The forest changes every day, telling us many different things. Meeting diverse plant life over 4 seasons. Studying the bountiful gifts of nature. A glimpse into the precious life of nature's monuments and hidden existence of rare species can be found here. The forest has become fertile and its value heightened. Designated by UNESCO in 2010 as a biosphere reserve, the nation's largest experimental forest research center. The nation's only research institute dedicated to the diverse species of the forest. Our nation's representative forest ecosystem report. It has become a resting haven for citizens, where the environment and culture harmonize as one. Think about the tomorrow that the forest brings us. A forest of healing that we seek out in times of need. A forest of hope to find the sounds of nature. A forest to bring us back to the beginning, to cherish nature's natural beauty. A treasured forest for our tomorrow. A space which is shared by the present and the past. Dreaming of a beautiful tomorrow, living in coexistence. We look forward to making a 1,000-year history at Korea National Arboretum.
Forests have endured a time beyond people's imaginations. Forests have numerous stories to tell. They are the habitat of countless lives and a place of rejuvenation for people. Let's meet the people who are Forest, where the Korea National Arboretum is located, has been protected as a royal forest since 540 years ago. Possessing a stunning natural environment, it is the habitat of life, ranging from rare plants to natural monuments. It's why the value of Kwangmun Forest was recognized globally and designated as a UNESCO biosphere. Kwangmun Forest is a natural masterpiece with a harmony of life, presenting unique and stunning scenery in each of the 4 seasons. It's a new morning at the Arboretum. The Korea National Arboretum is a special place where you can see the Kwangmun Forest and the various organisms that inhabit it. In 1999, the Korea National Arboretum was established to collect, preserve, and study the life forms of forests in a systematic manner. Today, it studies plants and insects that inhabit the forests, researches seeds and specimens, organizes exhibits, and provides forestry education. The Korea National Arboretum has organized specialized gardens which are open to the general public and experts by their use and taxonomical characteristics. The specialized gardens include the Ornamental Tree Garden, presenting a collection of trees with high ornamental value. The Flowering Tree Garden, which exhibits trees with beautiful flowers. And the Aquatic Plant Garden, Medicinal Plant Garden, Rare and Endemic Plant Archive, and Temperate House. The Korea National Arboretum also consists of specialized facilities for specialized activities. The Korea National Herbarium is the nation's first exclusive specimen facility, established to contribute to further research in related sectors through procuring large numbers of specimens, establishing accurate species categorization systems, and enhancing the usage of specimens through impeccable management and conservation. The seed bank is a mandatory facility for long-term preservation of the seeds, which are essential to the research, conservation, and usage of the plant resources. It is equipped with a high-tech management system for long-term preservation of seeds. The Tropical Plant Resources Research Center was established in 2009 in an effort to expand the scope of forestry research. It contributes the collection and preservation of tropical plant resources of infinite value, and to secure future resources which will allow us to prepare for environmental changes and heighten the value of plant resources. The Forest Museum is a place that exhibits the history and future of Korea's forests and forestry. It features various exhibits related to forestry, relics, and woodwork. The research team is climbing mountains to study the forest organisms of the Korean Peninsula. Today, the team is here to conduct an extensive study of the organisms that inhabit the Paektu Mountains. The researchers observe each flower and tree, reporting their number and characteristics. Monitoring the habitat on a regular basis is a crucial part of protecting our natural environment. The insect team and mushroom team are also collecting specimens. Insects and mushrooms are key constituents of the forests, and there is an extensive research regarding them. The efforts to protect the beautiful natural environment and biodiversity must continue until we complete the biological survey of the Korean Peninsula. Let's meet the preservation team who are working to protect Korea's endemic plants. The Korea National Arboretum is leading efforts to preserve life forms throughout Korea. Observing the habitat of plants and protecting endangered species are two of the Korea National Arboretum's key roles. Reviving endangered plants is also very important. Recently, the Korea National Arboretum successfully revived a rare orchid through proliferation. Each year, the Korea National Arboretum designates endangered rare plants and restores them in a systematic manner. Throughout the country, there are restoration sites that have been restored by the Korea National Arboretum. Since we can't bring back nature once it's lost, the Korea National Arboretum's efforts to protect this nature must continue. Forest organisms collected throughout the country are brought to the Korea National Arboretum.
Good afternoon, everyone, for ISGO 2020. Did you enjoy the lunch with Korean native folk plant? Here at the venue, we had a Korean traditional lunch, 구절판, the platter of 9 delicacies, which is 9 traditional food together in one dish. It was really good, and I hope you had great lunchtime all together. So now we are moved on session 2, ornamental exploration and utilization by Professor Chun Young-song. So I would like to turn the chair over to Professor Chun Young-song from Korean National College of Agriculture and Fishery for the following session 2. Thank you.
Okay. Thank you. Yeah, Professor Kim. I'm back as a chairman. The second session topic is ornamental exploration and utilization. We have 3 topics, 1 invited and 2 oral presentation. The first presenter, Dr. Ryan Contreras. Yeah, let me introduce him some in detail. Dr. Ryan Contreras is a professor of ornamental plant breeding in the Department of Horticulture at Oregon State University. There, he teaches spring and fall woody landscape plant identification, plant growth and development, and plant propagation courses. He established the directed ornamental plant breeding program, which focused on the developing new cultivar or woody shrub and trees. And his goal is for the breeding to directly support and enhance the success of nursery growers, landscapers, and consumers. Okay, the topic today, creating your own path, inducing double trait using mutagenesis. Okay, Dr. Ryan Contreras, please start your presentation. Do you hear me?
Yes. Do you hear me?
Okay, let's go.
Okay. Very good. All right. Well, thank you, Professor Song and Professor Kim, Professor Zhang, for inviting me. It is an honor to be invited to speak with you today. So, yeah, I'm going to talk about a number of different projects that we have that are ongoing to develop better forms of landscape plants. So in my program, we work on about 50 different genera, or close to that, to try to address a lot of the different issues in my industry. So I'm going to try to get through a lot in a that's available. Professor Zhang talked about apomixis earlier. I'm going to touch on that in my own presentation. And so in those cases, you need to induce some mutation or induce variation, and we use mutagenesis or mutations to do that. Just one quick example, we use all of these different techniques and lots more. But this is an example from Styrax japonicus where we combine the weeping habit of Marley's pink or pink parasol with the purple foliage of evening light to develop a purple weeping form of Styrax japonicus. So we do a lot of that as well. And this isn't new. Nothing that I'm going to talk about is revolutionary. I think we've just done a pretty good job of employing this. Radiation dates back to the 19th century and has been used in plant breeding since the very early 20th century. So this is not, it's not like I'm talking about CRISPR today. So this is not new, but it is relatively new, newer, I guess. It's been used more recently in ornamental plant breeding than it has in crop breeding. So this is typically, I just borrowed this from a paper from 2015. This is typically what you see, especially in crop breeding. This is sort of the flow through that you would see for using mutagenesis in a crop breeding program, where the mutagen of choice is applied to the original seed varieties. And then in the M1 generation, you get chimeras. So if a plant has 2 copies of a gene and you knock out or you alter 1 copy, it still has the other, and it leads to hidden recessive mutations, and therefore you must go to the M2 generation or the M3, 4, and so forth. Okay, but as again, Professor Zhang talked about the fact that woody plants have a very long generation time. It can take a long time to go multiple generations. Going to the M4 often in many of our crops is not really a viable option. Luckily for us, or luckily for me, we're often, most of the woody plants work on are highly heterozygous to start with. And so we actually see a lot of mutations in the M1 generation. So we actually, if we did go to the M2 or M3 generations, certainly we would continue to uncover more hidden recessive mutations. But I have been very pleased in our short amount of time doing this at how many mutations we're seeing in the first generation. So I'm going to try to get through a number of projects today, including an herbaceous perennial, a number of different shrub projects, and then touch quickly on some tree breeding work that we've just started over the past couple of years. In each of these, what I hope to do is to lay out, at least briefly, what the problem is, how I want to improve these plants, and then what we've done. And in each case, we've done some sort of mutagenesis, and then where are we in that project? And by this, I hope to show the benefit of these different techniques across a wide range of different taxa. The 2 methods of mutagenesis that I'm going to touch on are ethyl methane sulfonate, or EMS, and gamma radiation. Of course, EMS is available from any number of chemical supply companies, Sigma Aldrich or Fisher and so forth. And it's generally applied. It is a liquid, very carcinogenic, highly carcinogenic, so great care is required in safety protocols. But a soaking is done at some concentration for some amount of time, and then it is, this is not pouring EMS down the drain. I have poured the EMS off, and then the seeds are rinsed thoroughly for 15 to 30 minutes and before putting back in a container, taking to the greenhouse, and then sowing out. So it is somewhat cumbersome to deal with a highly carcinogenic chemical. Both the application, the cleaning of the seed, the disposal of the chemical, and so forth. But it's very relatively straightforward to apply. A buffer is required, but that's talk for another day. But it's relatively inexpensive. On the right is we have a gamma source here at our radiation center at Oregon State. And so we're able to just take these seeds over and I give them to a radiation technician who exposes them to our desired rates. This is very convenient because it's here on campus, so it's just a short walk to the radiation center. And it is convenient in that I give him the seeds, he gives them back to me within 30 minutes, and they are no longer radioactive, as opposed to when if you're using fast neutrons, for instance, the seeds remain radioactive for a period of time. So we're able to impose these treatments, and then within an hour, we can have them in the greenhouse and sowing them. Unfortunately, it is more costly. It's not terribly expensive for a single radiation dose, but it's $150 each time it goes down. So 150 US dollars. If we're doing 10 to 20 different treatments on a number of different taxa, it can get quickly into the tens of thousands of dollars US. And so we're running a relatively cheap budget program. So we balance the convenience of each of these with the cost. Okay, so the first project that I'm gonna go over is Cape hyacinth, or Ornithogalum candicans. So it is it's an herbaceous perennial. It's also known as Galtonia candicans. And the issues with it, there's issues with lodging. It has great potential to be weedy, or dare I say invasive, based on the amount of seed set that I've seen in some stands around Oregon. And overall, it's too tall for many landscapes. So we want more and more, the consumers are demanding that the plants be shorter. And also, when our growers are shipping herbaceous perennials, they want them to fit onto a shipping rack. So that's just simply that growers have asked us for. So we wanted to address these three issues, and especially lodging and the height is very closely related. This is the only cultivar that I'm aware of, Galtonia moonbeam. Oh, and by the way, I should mention this is this species is from South Africa, so it is actually highly drought tolerant, which is another desirable attribute of the species. But Galtonia moonbeam, it is improved because it is a double flowered form, but it is very tall. It requires some structure to hold up the scapes that flop over readily on the ground. This is a wild type, and you can see that when it's planted in mass, it holds itself up relatively well, but plants around the side, the scapes will flop over and lodge on the ground. It's an undesirable trait. So to address that, we exposed seeds to varying rates of EMS, 0, 0.2, 0.4, 0.6, 0.8, and 1.0. And we did this as a 24-hour soak, and we either did it in addition to a water soak or an imbibition period for 24 hours prior to EMS, or with no pre-soak. And you can see there was a quadratic response of seed germination when we did the 24-hour soak, but we quickly got to 100% mortality. So if one is going to use this 24-hour imbibition prior to exposure to EMS, then you would need to decrease the subsequent exposure time to EMS. Without the pre-soak, we observed a generally a linear decline in seed germination. But regardless, as we increase seed, the EMS concentration, we saw a reduction in seed germination. Very common response. Here are above, this is with the 24-hour pre-soak, and you can see, again, no germination at 0.6, 0.8, or 1.0% EMS. Below is directly into the 24-hour EMS soak. And again, a delayed and reduced germination with increasing EMS concentration. So after these seedlings emerged, we potted them up, grew them on for a while, and eventually observed them in our field plots. And here's what we saw. The two trend lines that we see here are from 2014 and 2015, respectively. And so over both years, we saw a reduction in scape height, we saw a reduction in lodging, and we saw a really dramatic reduction in average seed set per capsule, which was really exciting. So overall, the plants ended up being shorter, there was less lodging, and they were less fertile. And just showing you the actual raw data here. Again, in 2014 and 2015, we saw a decline in seeds per capsule with increasing EMS concentration. There was also a decrease in seed germination with increasing EMS concentration over both years. And then in 2015, we actually looked, because those seeds per capsule and germination really are getting at female fertility. We were curious if the same trend was going on with male fertility. So we at least did pollen viability in 2015, and you see from 83% in the control to less than 3% in the 0.4% treatment. So the plants are less fertile, both male and female. From those, we took, from that population that you saw previously, we took 10 plants, we dug them and put them in this row as superior genotypes. And here are a few of those. So these plants had, they exhibited no lodging, they exhibit no seed set, and they're generally floriferous. And we selected a number of different forms that had, so a little more petite on the left, more floriferous on the right, and not surprisingly, growers have really chosen the one on the right. And just showing you there is, they don't look good as we enter into October, but there's no seed set on these plants. And we have maintained seeing no seed set on these and plants propagated from them over a number of years. Flowering currant, or Ribes sanguineum, is native to my area. It's a great plant for early season pollinators. Great spring flowers. It's relatively drought tolerant, but after it's done flowering, it's not a very good plant. So we exposed it to a number of different rates of EMS. You see at the bottom here, 0.2, 0.4, 0.8, and 1.2. And we sowed these out in a greenhouse, and just showing you one example of the control versus a 0.4% that was treated for 24 hours. Again, great reductions in germination as we increased EMS concentration. We then grew these plants out in containers. And one thing I want to point out, I've really been concentrating, because we work on 50 different genera, we don't spend a lot of time on really minor mutations. And so I have not done a good job in any of these projects on quantifying minor mutation rates. So really there's a lot that's missed. We really pay most attention to gross morphology of foliage, of flowers, and of plant size and structure and form. But there are a lot of these interesting mutations, such as increased petal number, that for this plant is really not that critical. But it is an interesting observation that if I had 20 more students, we could really dig into perhaps. But this was the first mutation that we really saw. So the wild type is on the left, and my plant is on the right, and this arose from, I think, a 0.2% treatment. And so this deeply dissected leaf, we then grew it on, and it turned out that this was, we released this plant as Oregon Snowflake. It is this highly dissected leaf. It is much more compact and densely branched, and as overall is a better landscape plant. This is just showing you the leaf form of a plant in situ out in a landscape trial, and it is a white flowered form. And here is the original mother plant. It's been transplanted, but still very floriferous, still very densely branched, and a much more improved form over the wild type. We then wanted to see, could we use this new mutation in breeding? And indeed, it is heritable in the next generation. And so now we crossed it with different colors, and we now have a pink that has the dissected foliage and compact habit, and a red that has those same traits. So we're pretty excited about completing the series. In shrub dogwoods, so in Cornus, not the big bracted dogwoods, but in the shrub dogwoods that are very hardy for us here in North America, I've had growers who came to me and said they want more dwarf options that had improved flowering quality over the available dwarf selections. So we exposed a large collection of seeds to 0, 10, 25, and 50 kilorads of gamma radiation, and then planted them out, just showing you 25 and 50 kilorads of gamma radiation and then planted them out, just showing you what it looks like in our greenhouse after one of these treatments. And just again, quickly with the data, we had reduction in germination with increased gamma radiation. And we had a reduction in plant height in Cornus racemosa, in Cornus sanguinea, and in Cornus sericea. You will note that these plant heights are quite short. So these were still in the first year after treatment. So this is just, we take measurements in our first year so that we can start throwing plants away. And Cornus racemosa and sanguinea, the LD50 was between 10 and 25 kilorads. Sericea was less than 10 kilorads. And so we've reduced germination and height following gamma treatment. And here's what the full study looked like. So the plants with the orange flags in the foreground were selected from this large population in the background. So quickly roguing down this population, and we'll be down, probably throw away 90% of these plants in the next 2 years. Pyracantha, I'm interested in pyracantha as a boxwood replacement. I think it could provide multiple season interest as an improvement over boxwood, particularly in the United States. We have issues with boxwood blight. So we exposed plants to 0.1, 0.2, 0.4, and 0.8% EMS. And we observed an LD50 of approximately 0.2% EMS. We did measure 1,500 plants that were in 1 gallon containers. I haven't had that data entered yet. But of those, we field planted 13 plants, and 6 of these compact selections we have propagated for advanced trials. And I've put those numbers in red parentheses here. So 3 were from the 0.1 and 2 were from the 0.2. 1 was from the control, showing you that indeed you do get some seedling variation, which it's always important to separate. Is my treatment having an impact, or is it just simply seedling variation? Fragrant sweet box, Sarcococca confusa. So this is a great plant. It's evergreen. It tolerates dry shade. It has clean foliage. It's fragrant in the winter. It's non-rhizomatous, but I want it to be more compact. Here's showing you the flowers and great fruit of Sarcococca. But again, as our colleague from the University of Georgia mentioned earlier, polyembryony is a real challenge in breeding. And so you can get up to 4. We've had up to 5 actually embryos in a single seed. And so you don't know which one is actually going to show variation. So we expose those seeds to a number of different rates of EMS for 24 or 48 hours, and just showing you that in the circle icons are germination. So in both 24 and 48 hours, we reduced germination with increasing EMS. What was really interesting to me is we also, particularly obvious in the 24-hour treatment, we saw a reduction in twinning. So that is and I'm using twinning as a proxy for polyembryony. So as we increased our EMS concentration, we reduced the amount of polyembryony, thus allowing us to more effectively breed and select Sarcococca. So we grew these plants out in containers. We then field planted them of size, watched them for a while, and this is exciting to me because, going back to my first slide, really all plant breeding is variation. And you look at the size of the variation in this 1.2 for 24-hour treatment. And that's really where we can make significant gains in plant breeding. We had a number of different variations here. We had a number of different compact selections. There was a chlorophyll mutant, lots of different leaf mutations. Ultra dwarfs, and so this is an exciting ongoing project. Very slow growing species, so difficult to say too much at this point. Spirea douglasii, this is another one that's native to my region. I love this inflorescence. It's dramatic and somewhat unique in the genus. It seems adaptable in my observations. We see it in ditch banks, in very wet areas, but I've also seen it in more upland locations, and great opportunity for an adaptable shrub. But it suckers from the base and it forms very thick, dense colonies, and so we wanted to improve that. And so we expose it to a number of different rates of gamma, including 5, 10, 20, and 40 kilorad. This was very tolerant to gamma radiation. Really, our LD50 was not until we reached 40 kilorads, and we reduced the plant height once we reached 40, but there was really no impact up until that point. And so far, these are two plants that are now 3 years old, I believe, and we have not seen any suckering in them. That doesn't mean they're not in the next year or two, but thus far we have seen no suckering. So very excited about these plants. And we did collect seed off of these this year to go to the M2 generation. All right, I know I'm approaching the end of my time. Trees, we need more options. We have terrible monocultures in our urban forests, and we need more options. We are overplanted in maples, even though I'm still breeding maples. We need more options. I'm just showing you here's an example in the eastern United States of catastrophic losses from, in this case, from Dutch elm disease, but we've had emerald ash borer, and we really need to diversify our urban canopy. So with that in mind, we have done mutation on Celtis, Cercis, Cotinus, Philodendron, Quercus, and Zelkova, with more and more to come. So, and just a few quick observations. In Cercis occidentalis, I want an upright form that we can start breeding with Cercis canadensis. And we had very poor germination in every treatment, including control, but our approximate LD50 here was 0.5% EMS. And it's hard to say again, but it does look like we've reduced the plant height in Cercis occidentalis. Cotinus cogagria, I'm really interested in also in a fastigiate form. Again, terrible germination in each of these, but it does, we did clearly reduce germination as we increased our gamma radiation from 10 to 25. So we're really between 10 and 25 is where I think the LD50 will be on Cotinus. And but we had poor survival above 10 kilorads. I want to just point out the methods on the trees quickly to end. These were, we did our treatments and then we lined them out because trees are very difficult to grow in containers. So we lined them out on 60 centimeter centers, observed them for 2 years, made selections, which are the orange flags, and then these were spaded and put on 3 meter centers for a many year observation. Last couple of slides, Philodendron amaranths. Just poor germination all the way around, no discernible LD50, and this is the way science goes sometimes, that our 0.8% treatment was actually taller than our control. Can't explain that. And then Quercus, we had 98% reduced to 13% germination in the 0.2 EMS treatment. So that's a substantial reduction. Quercus robur fastigiata, we had we didn't observe our LD50. We only did one treatment of 0.3% EMS, but we did observe some reduction in plant height. So my very fast rambling in all those different taxa is that we have observed effective non-targeted mutagenesis across a broad range of taxa using EMS and gamma radiation. And yes, the selections are ongoing, and we are going to future generations, particularly in the shrubs, but even in the M1, we have seen substantial variation, and I'm really looking forward to the future generations with increased variety. And I appreciate your time and attention, and again, it's quite an honor to be able to speak with you, and I wish I was there seeing each of you. So there may be time for questions, I'm not sure.
Okay. Thank you very much for your presentation, Dr. Ryan Contreras. Okay, have any question from floor? Okay.
Thank you, Ryan. I have one question about your presentation that you mentioned that normally EMS treated material after germination, it is growing very slowly and being dwarf, right? As my experience in other cases, normally gamma ray treatment or EMS on other material, that in the beginning of after treatment, in the beginning of 1 or 2 years, it comes very dwarf and very slow growth habit. But after that, maybe 3-4 years later, it recovered totally and grow as a normal stage. What do you think about?
It's a great point, and there are some of these projects. Well, I've only been at Oregon State doing this for 10 years, and some of these projects are only a couple of years old, particularly the trees. And I fully expect that in many, probably most of the examples that I showed on the trees, that yeah, I think that would fully reasonable that they're going to recover. Some of the other projects, so like ribes, that's been going, that's an ongoing project from about 8 years, and they haven't recovered. The Sarcococa, they're just notoriously slow. Wild type is very slow. And I've been watching those plants for about 7 years now. But yeah, it's a great point. And it's very difficult to say much with great confidence after 2 or 3 years. So yeah, long-term evaluation is going to be needed.
Okay, thank you for the answer, Dr. Ryan Contreras. Okay, time to move next topic. Okay, next presenter, Dr. Jae-hyun Kim from Korea National Arboretum. The topic is development of mass propagation method for the Viburnum terratanum using seed and vegetative propagation. Okay, Dr. Kim, please.
Good
Start.
afternoon, ladies and gentlemen. I am Jae-hyun Kim, Research Institute of the Korean National Arboretum. What I will be presenting the development mass propagation method Viburnum dilatatum using the seed vegetative propagation. Viburnum is a genus of the 150 species of plants with showy flowers. They are spread all over the world and very popular for gardening because of their beautiful flower and fruits. Securing materials is very important for the industrialization of useful plants. There are two methods for securing materials, seedling and vegetative propagation. In the case of seedling propagation method, a large number of plants can be obtained within a short time, but it is difficult to break the seed dormancy. Many have classified seed dormancy since the past, but the classification that has recently been accepted as orthodoxy has been categorized the Veskin group. The Veskin group divided the dormancy of the seed into 5 broad categories. Physical dormancy that does not absorb moisture due to seed coat. Morphological dormancy with undeveloped embryo. Physiological dormancy due to bioactive substance. Morphophysiological dormancy is combination of the morphological and physiological dormancy. And combinational dormancy is combination of physical and physiological dormancy. If germination occurs after more than 30 days at the germination temperature, it is determined that there is a physiological dormancy, and the dormancy can be overcome by cold or warm temperature stratification. We developed the sexual propagation method and the asexual propagation method through 2 experiments. The sexual propagation experiment was experiment with IBA. A type of auxin was treated by concentration and development of rules according the cutting media. In the sexual propagation experiment IBA, concentration of 0, 10, 100, 1000, and 2000 milligrams per liter were used, and the treatment time was immersed for 10 seconds. As per the cutting medium, 5 types were used shown above. This is a result of a sexual propagation experiments. It can be seen that the difference according to the concentration of IBA is not rising. There was no difference between control and other treatments. This is a numerical representation of the previous result. This graph showing the survival rate and the looting percentage. As with period, there is no significant difference between each treatment. Based on the result, it can be determined that the looting of the viburnum dilatatum cutting was not affected by the IVA treatment. There are the experiment results for each cutting medium. As shown in the picture, the routing was the best commercial medium and Kanamazuchi mixture treatment. This is a graph showing previous result in numerical terms. Like a result in the picture, it can be seen that the highest routing rate and survival rate were shown in the commercial medium and Yamaguchi mixture treatment. This result means the medium with a good moisture and drainage is advantageous when cutting viburnum dilatatum. Next is a sexual propagation experiment. Experiment was performed according to the characteristics to the each dormant, and physical dormance was determined by imbibition testing. In the case of the morphological dormancy, we started through the growth of the embryo, and the physiological dormancy was examined through the temperature experiments. In addition, we investigated the details of dormancy by move-along testing. Move-along test is experiment to observe the pattern of seed germination by artificially controlling its change of the season. It was divided into temperature fixed control and the temperature change, a seasonal change. The temperature change was divided into treatment starting from winter temperature and treatment starting from summer temperature. This is a result of water absorption testing. In general, if the weight increased by more than 20% relative to dry weight with 12 hours, it is judged that there is no problem with moisture absorption. Therefore, viburnum dilatatum can judge it there is no physical dormancy because there is a problem with water absorption. These photographs show the process of seed germination by viburnum dilatatum seed. It can be seen that the embryo grow and the germination and cotyledon come out. Since It can be seen the size of embryo grow from times of dispersal of germination. Viburnum dilatatum is considered to have immature embryo, and thus it can be considered that it has a morphological dormancy or a morphophysiological dormancy. This graph showed germination rate by culture temperature. Germination started on the 16th at the 20 degrees seed treatment, and the germination rate of more than 85% was recorded in the 3 days. In other temperature treatment, rich germination occurred. Viburnum dilatatum was judged to have physiological dormancy because it took more than 15 days or 20 degree. Seed treatment with germination most quickly. Looking at the previous result, comprehensive Viburnum dilatatum is judged to have a more physiological dormancy. In morphophysiological dormancy, there are thus 9 subclasses according to each characteristics. To investigate the subclass Viburnum dilatatum, since move along test was performed. This graph is the result of the move along test. The red arrow is the point at which the temperature was changed. In the case of some stars, it can be seen that germination has started after a high temperature period. In addition, it can be seen that seeds have undergone high temperature, germinate rapidly, and germinate more than 80% in 4 weeks after germination begins. In the case of winter star, seeds that have undergone low temperature do not germinate, but it can be seen they are germinated experiencing high temperature of 30 weeks. Therefore, it can be seen that Viburnum dilatatum's seed need a high temperature to grow embryo and high temperature required to break the dormant seed. Talking above results together, it is judged that Viburnum dilatatum seed have non-deep simple morphophysiological dormancy, or non-deep simple epicotyl morphophysiological dormancy among morphophysiological dormancies because it needs high temperature to break dormancy and high temperature to grow embryo. Although the results were not added due to time relation, it is found that there was epicotyl dormancy through additional experiments, and as a result, it is found Viburnum dilatatum seed has dormancy of non-deep simple epicotyl morphophysiological dormancy. On the map, it can be seen the type of seed dormancy of viburnum plants spread all over the world is similar. In general, morphophysiological dormancy is known as the oldest seed dormancy type. It can be seen that the seed dormancy patterns of the viburnum plants are well preserved and do not change as much as they evolve. This picture is the pole of the Korean National Arboretum, where I belong. Autumn in Korea is so beautiful because of the autumn leaves. If you have a chance, I recommend you to visit in Korea autumn. Thank you for your attention.
Okay. Thank you for the presentation. Any question? Okay. Thank you for the presentation. We don't have enough time to Q&A. How about move to next topic? Okay. Next topic is the effect of water stress on energy and the growth of glacier bicapsulas. The presenter, Dr. Fei Yong Liao from Central South University from China. Okay. Give a hand. Okay. 이거 했는데. Okay.
See the picture.
Okay, please.
Okay. Dear Chairman, I'm Liao Feiyong from Central South University of Science and Technology. My report is the effect of water stress on the energy utilization and growth of Cassia bicapsularis.
还有是学霸嘞，当时在考试，学习能力很强。
Water is a necessary elemental factor for plants. Water content in the soil affect plant growth and even lead to death. Cassia bicapsularis is a shrub of Cassia genus. Grown from around October, and it could be enjoyed for 2 months from September to November. We have preliminary research on the effect of water on its physiology. The blood showed that the plant could endure 6 months waterfront of growth. The plant was affected in the leaf. The plant was affected in the leaf of color and shape. While it was not clear how plants adapt to flooded element from the perspective of light utilization and its energy utilization. In order to make rational use of Cassia bicapsulari in the garden, the effect of different soil water content on the energy utilization and growth of Cassia bicapsularis was studied in order to find out its adaptive machine and guide its application in gardens. Experiment materials. The 2-year-old seedlings of Cassia bicapsularis were planted in the place in the campus of Central South Forestry University of Science and Technology in Changsha, China. The temperature and the humidity was natural. The treatment of soil water content was divided to 5 groups. They are CK, WS, SD, MD, and HD. Determination content and method. The soil water content was measured by TDR 100 The length of FDR sensor was 20 centimeters. The geography concentration, various parameters, net cover and AC cover, leaf biomasses, and MDA were measured. Result and discussion. Effect of geographic content. After treated for 20 days, the total geographic content of HD increased slightly, but others decreased. That of MD decreased mostly. The total geographic content of MS decreased gradually with the extension of the flooded time. Only 74.79% of CK are treated for 71 days. The total geographic content of MD was lower than that of other treatments. Effect on FO. After treated for 20 days, the FO of MD was 76.68% of CK, while that of HD was 1.04 times of CK, but the difference was not significant. Effect of FV flash FM. After treated for 20 days, the FV flash FM of HD was 67.19% of that of CK, which indicated that the brains of HD had been seriously stressed. The difference of other groups with CK was small, and the difference was not obvious by statistics. Effect on QP. After treated for 20 days, QP values of HD decreased to 60.59% of CK. That of MD was 1.09 times of CK. QP values of MD and SD were higher than that of CK. Effect of QP. The QP values of WS increased first and then decreased, which indicated that long-term treatment led to the decrease of the utilization efficiency of net energy. After treated for 20 days, ETR values of HD was 40.57% of CK, and those of WS, SD, and MD treatments were similar to that of CK. Effect on VC max. VC max of WS was 49.08% of CK after treated for 71 days. Its change indicates that the VC max gradually decreased with the extension of flooding time. VC max of SD increased gradually. After treated for 19 days, VC max of MD was 65.32% of CK. VC max of HD decreased first, then increased. Effect on photosonic rate. After treated for 20 days, the difference among the treatments were obviously. The photosynthetic rates of WS and MD were 69.26% and 74.65% of CK respectively. Effect on respiratory rate. The respiratory rate of SD and MD increased after treated with 19 days, but that of MS increased after treated for 71 days, which was 143.5% of CK. After treated for 20 days, the water content of WS, SD, and MD were increased, but was not obviously by statistics. while that of HD decreased significantly. Effect on leaf biomasses. The biomasses per unit leaf area of treatment increased after treated for 20 days. That of MD was the largest, then that of HD. Effect on plant morphology. After treated for 10 days, a lot of adventitious roots of WS plant began to grow at the base of stems above the ground. After treated for 71 days, the number of the adventitious roots increased continuously in the water. After treated for 60 days, the plant of WS grew well. Many of the adventitious roots floating on the water surface. The plant of WS still grew well after treated for 90 days. After treated for 20 days, the plant of SD and MD grew well. After treated for 17 days, the plant of HD began to wilt. After treated for 20 days, the plant died. The effect of water-fronting stress on light energy utilization. After treated with the water-fronting for 71 days, the changes of physiology parameters indicate that the energy absorbed and transferred decreased with the extension of fronting time, but its fixed energy is still more, indicating that the plant could adapt to fronting element, but the long-term fronting affected its growth. The effect of light drought stress on light energy utilization. The above analysis showed that the plant was not stressed by light drought treatment for 20 days. Those change of parameters indicate that the plant increased its capture of light energy, energy transfer, and energy fixation, and at the same time reduced its energy consumption, resulting in the increase in biomasses. Night drought had little effect on the plant growth. Therefore, the night drought could not only ensure the plant growth, but also save irrigation cost. Data showed that after treated for 19 days, the growth of plant was inhibited. The energy absorption, transmission, and fixation decreased, but the leaf biomasses was still more, which indicated that the plant was able to tolerate the stress of moderate drought. The plant shape also proved it. Therefore, 20 days of moderate drought was the tolerable range of plant. This indicated that the drought changed the conservation, transferring, and fixing of energy, but in a short time, it promotes the accumulation of leaf organisms to adapt lack of water supply. After 12 days of drought, it had a great influence on plant. That indicated that treated for 20 days, the growth of plant of SD was inhibited. Those changes indicated that the decrease of energy absorption, transmission, and fixed energy, especially the stopping of respiration, which indicated that the death of plant. Therefore, we have the conclusion. First, the plant could endure the fronting stress for more than 19 days. After 71 days of fronting, there were many area roots emerged from the stem base and floated on the water surface. The second, night drought had little effect on plant growth. It could be used to reduce the maintenance cost. Third, moderate drought affected the plant. It significantly inhibited the growth of plant after 19 days, slightly decreased the energy conservation, transmission, and fixation, and increased the energy consumption. Plant died after treated with heavy drought for 71 days. When treated for 19 days for 7 drought, the growth of plant was seriously threatened. 5. 18% to 25% of soil water content was the best water content of Cassia bicapsularis in garden green spaces, which could reduce the irrigation water and could not affect plant growth and ornamental. That's all. Thank you.
Dr. Fei-Yong Liao, have any question on the floor?
Your voice is more, I can't hear clearly.
Sorry?
Your voice is small.
Huh. Mm-hmm. Okay, thank you very much for your presentation, Dr. Liao, Fei-Yong Liao.
Yeah?
Okay,
thank you.
Time to
Thank you
finish.
for your attention.
We have break time, 30 minutes. Okay, have a good time. 15?
Hello everyone and online audience. My name is Wan-Sun Kim from the University of Seoul, South Korea. Actually, today I'm served the chair of the second half of session 2. On the list, there is a chairperson is Professor Im Jin-hee, but she is not available at this moment, so I substitute her. Actually, first of all, as you guess, I am an editor of the symposium, Acta Horticulture. So at this times, I'd like to thank you, all of you who contribute and supported to Acta Horticulture publication. Thank you very much. And also, you can access the brand new Acta Horticulture in the website of ISHS. So the Acta number is 1291. You can find it. All right, I'd like to move to the first presentation. The title is Abscisic acid sinensis inhibitor inducing flowering of dormancy peony without bud abortion, which is presented by Professor Yong-ha Lee, Professor Seung-yeon Lee, and Professor Ki-sun Kim. Actually, the Professor Lee graduated the Seoul National University and got the PhD degree at the same university. And now he's working at the Daejeon University, the central area of Korea. So, hi, Professor Lee.
Thank you.
Okay, I would like to invite him for his presentation with a big hand.
Good afternoon, everyone. I'm Yong-ha Lee in Daejeon University. Today's presentation is one of the studies I did during my doctoral course in Seoul National University. This is my topic, ABA synthesis inhibitor inducing flowering of dormant peony without bud abortion. Peonies have many species with flowers of various colors and shapes, so they have been used as co-flowers for many years. However, the flowering period is very short, from May to June. So, up to now, there are many studies to extend the flowering of peony. Our study also focused on the off-season culture of peony. This picture shows the peony rootstocks having dormant buds. For breaking the dormancy of the buds, 3 or 4 months of cold temperature treatments were needed. This cold temperature treatment could promote the flowering of peony. However, this cold temperature treatment required a long period of treatment and need facility, such as a large cold room chamber. Gibberellic acid plant hormone could be an alternative method to promote the peony without a long period of treatment. There are some trials applying GA on peony. In 1990, Evans treated GA on peony tubers, but unfortunately, all flower buds aborted, like these pictures. And in Chang's study in 2009, flower bud abortion was also observed when peony was treated with GA 2000 ppm. This is another study in 2012. In the peonies subjected to sufficient cold temperature after December and January, GA treatment could break the dormancy and produce the flowering normally. However, in the peonies subjected to insufficient cold temperature before November and October, GA treatment could break the dormancy, but But within about 2 weeks, all flower buds aborted and shoots blasted, like these pictures. So I wonder why, when the GA was treated in uncooled peony, the plants withered and flower bud abortion occurred. This is GA and ABA balance theory. This theory means that the dormant state or non-dormant state is regulated by the balance of GA and ABA contents. In the dormant state of the seeds, the genes that synthesis ABA and degrades GA is expressed at a high levels. So the ratio of GA and ABA is low, decreased. When the seeds were subjected to cold temperature, naturally and artificially, the pattern of gene expression is reversed. So the genes related with GA synthesis and ABA degradation is expressed at a high level. So the ratio of GA and ABA increased. So this high ratio of GA and ABA promote the germination of the seeds. Actually, this theory was made to explain the dormancy cycle of the seed. For explaining why the peony died back when treated GA, I applied this theory to the peony. I made to this hypothesis. Before winter, in peony rootstocks, GA synthesis decreased and ABA synthesis increased. So the ratio of GA and ABA is low and maintained to the dormant state. During the winter, for about 3 months, the pattern of gene expression is reversed. So GA synthesis increased and ABA synthesis decreased. So the ratio of GA and ABA increased gradually. So this high GA and ABA ratio promote the flowering normally. However, without cold temperature, naturally or artificially, I thought that the manner might be different. In dormant peony, the ratio of GA and ABA is low. When the GA was treated externally, the GA content in peony suddenly increased. So the ratio of GA and ABA increased temporarily. However, at this time, the peonies was confusing that the dormancy was broken and produced these shoots and leaves. But because these peonies had not been subject to cold temperature, the gene expressions would decrease in GA, but increased in ABA. So the ratio of GA and ABA is decreased gradually. Eventually, I think I thought that this abnormal hormone balance could cause the shoot blasting and flower bud abortion. I designed the experiments. As discussed before, there is if the peony did not subjected to cold temperature, there is a problem that the ratio of GA and ABA is low. So I thought of 2 ways to solve this problem. The first way is using fluoridone. Fluoridone is one of the ABA synthesis inhibitors. So fluoridone would prevent the ABA synthesis. As time goes by, the GA and ABA increase. The second way is to spray GA repeatedly. I think this treatment would help keep the GA at a high level over time. So we obtained uncooled peony, and there are five treatments. The first treatment is drenching distilled water as a control. The second treatment is drenching 100 ppm GA. The third treatment is 500 ppm GA drenching. And the fourth treatment is GA and fluoridone mixed drenching. And the fifth treatment is drenching GA and then spray GA repeatedly every 2 weeks until the end of the experiment. I was expecting these last two treatments would have a good result. In result, in control, no sprout occurs until the end of the experiment, so there was no flower and shoot. But when drenching GA on peony, sprouting was observed in all treatments, and there was no significant differences between hormone levels in number of shoots and plant heights. However, only 2 treatments of the fluoridone and GA spray could promote the flowering normally by 85% and 22% respectively. This picture showing the percentage of shoot blasting. The treatment drenching with GA 100% ppm and GA 500 ppm. This treatment resulted in shoot blasting more than 80%. But the treatment of fluoridone and GA spray decreased the shoot blasting to 0 and 20%. This picture was the 15th week after treatment. A is treatment of soil drench with only GA 100 ppm, and B is GA and fluoridone mixed treatment, and C is treatment of GA spray. You can see the treatment of fluoridone and GA spray promote the flowers normally. In conclusion, by the treatment of fluoridone and GA treatment, the problem of flower bud aversion and shoot blasting could be solved. Okay. Thank you for your attention.
Thank you very much, Professor Lee. It is time for question or suggestion and comment. So now we are open the floor and online audience for the question. Is there any questions and comment? If so, I give us some short question on your presentation. Actually, you used fluoridone, the chemical, which is abscisic acid synthesis inhibitor, is explained. But actually, we know this chemical, which is herbicide. Actually, in general, we use this chemical as herbicide. So, didn't you have any negative side effect of this chemical to spray or drenching to your plants?
Yes.
If you have some idea or question.
Yes. Thank you for the question. As chairman's comments, there was a problem using fluoridone. Next page. This picture is using the fluoridone. When the fluoridone was treated, the flowering of peony was done normally, but the color of leaves became lighter and leaf closes progressed after treating fluoridone. This is the biosynthesis of ABA from GGPP and to ABA. When fluoridone does not block the downstep of the biosynthesis ABA, rather the block upsteps that produce the carotenoid. So the action of fluoridone could make the great damage to the overall plant growth, I think.
So you mean the concentration of that chemical is very important,
yes,
to apply to the plants, right?
Yes, I think if it will be possible to use PCR commercially, the other chemical compounds must be developed.
Okay, thank you. Any question? Okay, time is up. So thank you very much, Professor Lee. Your presentation is very informative. Thank you very much.
Thank you very much.
Now we are moved to the second presentation. The title is Identification and Characterization of phenylalanine ammonia lyase involved in biosynthesis of floral scent in Prunus mume, which is presented by Chinese scientists, the Teng Xuan Zhang and Qi Jiang Zhang and Fei Bao and Anqi Ding, Yang Zhuan Yang, Jia Wang, and Tan Gran Cheng. So they're from the Beijing Forest University. So Ms. Teng Zhuan Zhang, can you hear me?
Yeah. Oh, sorry.
So would your microphone volume is up a little bit? It's okay?
Oh, it's okay.
All right.
Hi,
If you're
can
ready,
you hear
we'll
me?
just start. Okay.
Okay. Hi. My name is Chengqun Zhang. My tutor is Qi Xiang Zhang. We come from Beijing Forestry University in China. Today, my presentation is about identification and characterization of phenylalanine ammonia lyase (PAL) involved in biosynthesis of fluorescence in Prunus mume. Sorry. Sorry. Fragrance is a significant trait. Is a significant and important character of horticultural plants. Fluorescence can attract visitors and provide pleasure. It also is one of the most significant traits in plant-animal communication and attracting pollinators. Fluorescence is consisted of a blend of low-white volatile organic compounds, most of which are distributed into three classes, including terpenoids, benzonoids, and fatty acid derivatives based on the biosynthesis. Fluorescence is a significant trait of Prunus mume, distinguished with other species of Prunus, and its fragrance is dominated by phenylpropanoids or benzonoids such as benzoic acid, benzoyl alcohol, eugenol, and cinnamon oxide and cinnamon alcohol. The formation of benzonoids and phenylpropanoids share the common substance cyanic acid, to which deaminates PHE. The reaction is catalyzed by a well-known PAL that is considered to the first enzyme of FVP pathway. However, there are later reports about characteristics and function of PALs in Pronos movement. Thus, we screened two PALs named PMPAL1 and PAL2 in maize genomic. In Table 1, Table 1 shows their features and sequence characteristics of them. Although the genomic sequence all contained two exons and one intro, their gene structures were different. To exploring the evolutionary relationship between PMPAL, Pronos movement and other species, the phylogenetic tree was constructed. I'm showing this figure. PLs from all species are clustered into two groups, Group 1 and Group 2. Group 1 and Group 2 will consist of PLs from dicotyledon and monocotyledon respectively. PLs from Rosacea species will cluster into Group 1, and the distance between PMPAL1 and PMPAL2 was faster, and they were clustered together with relative PLs from Rosacea species. Spatial temporal specificity was the most significant trait of the emission of fluorescence. Based on this, the expression pattern of PMPALs were explored using RNA sequence databases of different tissues and flower and different development stages. PL1 and PL2 are expressed in all tissues. PL1 expressed at a high level and leaf, root, and stem, while PL2 expressed under the highest level in flower bud than other tissues. Generally, the expression levels of PAL1 and PAL2 will range from budding stage to flowering stage, while the upregulation degree and RFPAKM value of PAL2 was more than PAL1 until flowering stage. Thus, we suggested the results suggested that PAL2 might contribute to the fluorescence of Pronos movement. Then we cloned them from the strong sensitive Pronos movement Sanlunvidie, named PMPAL1-SE and PMPAL2-SE. The protein sequence all contained the conserved significant sequence in the blank box. To determine functional activities of PMPAL genes, angry butterworm with recombinant plant stem were transiently transformed into the leaves of tobacco. The absorbance of cinnamic acid was measured using a spectrophotometer to show the catalytic activity for converting PHE to Ca. The result shows that the enzyme activities of protein overexpressing PAL1 and PAL2 can catalyze was far more than that overexpressed empty vector, and there were no obvious differences between PAL1 and PAL2. The senses and emission of lifestyle can affected by many aspects, such as transcription factor, hormone, and light. Thus, based on the genomic database, we search through the promoters. The table 2 shows the sense-acting elements of them. We found that the promoters can contain the sense-acting elements in response to light, hormones, and MYB transcription factor. In summary, 2 PMPAL genes work screened in the maize genome, named PMPA1 and PMPAL2. Phylogenetic trees showed that these 2 PALs clustered together with other PALs from Rosacea species. 2 PMPAL genes were cloned from strong sensitive pronous movement 三轮玉蝶。PAL1 and PAL2 could catalyze PHE to CA in tobacco leaves, while expression profiles indicate that PAL2 might play an important role in FVBP biosynthesis. Promoter analysis showed that many sense elements responsive to the light and hormones presented in the promoter regions of PMPALs. This work will lay a foundation on clarifying the molecular mechanism of phenolpropanoids or benzenoids pathway in pronous movement. That's all about my presentation. Thanks for your attention.
For your presentation. And
Hello.
Now it is time for question. Okay, it is not for me. Yes, yeah. Now, okay, Zoom is working. It's now. So it is time for question. Is there any question or comment on this presentation? Anyone? Or a comment? Okay, I give you one easy question to you. Can you hear me?
Hi.
Okay, can you hear me? It's okay?
What? Sorry.
Okay. Maybe she can have some connection problem by internet. Can you hear me?
Oh, I can't understand what you say.
You cannot understand. Maybe the connection has problem. So at this time, we cannot discuss on this presentation. So if you the audience wanna to talk with her, so just email to her or give a message. All right. So okay, thank you, Ms. Zhang. Thank you very much for your presentation and you're sharing your time. Okay, we have some saving the time. The next presentation is the last one for today. So we have only one presentation left. The presentation is stomatal and non-stomatal limitations of Rosa hybrid pink bell under drought stress, which is presented by Professor Lee Yun-si and S. He and Ji Wang and Wan-Sun Kim. Actually, the presenter, the Professor Lee Yun-si. Hi, Lee Yun. Can you see me? Okay. Actually, she graduated master and PhD degree at the University of Seoul under my supervision. So she's my student. Now she is working at the Henan Agricultural University. So I'd like to say hello to her. Hi.
Hello. Hello.
Good to see you again. Yeah, long time
Yeah,
no
nice
see.
to meet you.
Okay,
Long
thank you
time
for
no
your
see.
giving me a presentation today. Okay, so can you hear me?
Yeah.
Okay. Well, when you're ready, you can start your presentation. Okay.
Yeah, I'm ready. Okay.
All right.
It's so glad to see the chairman, dear Professor Kim, and it's an honor to be here to do this presentation. I am Shi Liyun from College of Landscape Architecture and Art, Henan Agricultural University. And the title of my presentation is Stomatal and Non-Stomatal Limitation of Rosa hybrida pinkbell under drought stress. I divided my presentation into following 4 parts. First, introduction. As we know, rose is one of the most important commercial flower crop in the floriculture, and it was considered as the gift of lovers, sold year-round for flower delivery. And it grown for cut flower production in greenhouses produce flower in flushes years around. So since the cut rose is considered one of the most attractive and economically profitable ornamental flowers, most rose farmers try to increase rose production by extending the day lengths through supplemental lighting. However, the growth under supplemental lighting requires irrigation. Otherwise, the growth rate is negatively affected by water deficit. From the previous study, we can see that when vegetative growth began to switch into flowering, significant reduction occurred in the shoot growth rate due to the drought stress. And the supplemental lighting contributed substantially to the photosynthesis during the night. However, in the daytime, it caused a negative effect, during which irrigation played an active role to compensate this negative effect. So from the previous study, we can see that the nocturnal supplemental lighting can increase the shoot growth and the quality. In the same time, it also increased the nocturnal transpiration and water loss. So without irrigation, the drought stress occurred. The restriction on plant growth from drought stress is largely imposed by the decline of the plant carbon balance, which is mainly determined by the photosynthesis. However, there is an ongoing discussion about whether the limitation factor for photosynthesis during the drought stress is stomata factors such as stomata closure and diffuse resistance, or non-stomata factors such as metabolic impairment. So the aim of this study was to understand how supplemental lighting and the drought stress it causes affect photosynthesis and what stomatal, biochemical, and photochemical factors are involved in this response. Furthermore, provide greater understanding of the response of cut rows to drought stress and the information needed to find out the appropriate irrigation practice. The next part, materials and methods. The cultivar pinkbell and the high-pressure sodium light were used in this experiment, and the greenhouse was settled as following. The plant were divided into 2 supplemental light treatment groups, the control and the supplemental lighting treatment group, PAR90. The supplemental lighting was programmed to come on between 4 p.m. to 0 o'clock and 2 a.m. to 10 o'clock, creating a nighttime of only 2 hours. Since no difference was found between the irrigated and non-irrigated regions,
under the control treatment group. So the two irrigation regimes were only applied to the supplemental lighting treatment group. The no irrigation and the irrigation once per hour during the supplemental lighting. The results and the discussion. From the table 1, we can see that in the supplemental lighting treatment group, rose plants flowered 6 days earlier than those in the control treatment group. Supplemental lighting without irrigation had a negative effect on the flower ornamental value, such as shoot length and petal length. The table 2 shows the photosynthetic status of the rose plants at a moment in the day and in the night. Compared to the control, plant treated with supplemental light but without irrigation had a 20% reduction in leaf relative water content. In the daytime, the night photosynthetic rate in the control and irrigation region were not significantly different. But supplemental lighting without irrigation caused a significant decrease in photosynthesis rate. In the night, the control treatment group showed a lower photosynthetic rate compared with the supplemental lighting treatment groups. However, without irrigation, it showed a reduction in the photosynthetic rate, stomata conductance, transpiration rate, and water use efficiency. The figure 1 shows changes in the diurnal photosynthetic characteristics. A twin-peaked diurnal pattern occurred under the control treatment group. The photosynthesis was at or below zero at night from 5 a.m. to 7 p.m. Nocturnal photosynthesis occurred under supplemental lighting treatment groups. The non-irrigation region showed a similar twin-peaked diurnal pattern at the control treatment group. However, the photosynthesis rate was significantly lower from 8 o'clock to 12 o'clock. The irrigation region turned the two peaks into one that significantly higher than that of non-irrigation region. No significant difference can be observed in stomata conductance between the control and irrigation region at most times. The lowest stomata conductance value occurred under the non-irrigation region from 10 a.m. to 5 p.m. And the non-irrigation region showed higher intercircular conductance levels in the afternoon from 2 p.m. to 7 p.m. compared to the irrigation region. And the non-irrigation region groups demonstrating the lowest stomata limitation from 2 p.m. to 7 p.m. So from the result in table 2 and the figure 1, it can indicate that the plant under the drought stress exhibited a significant reduction in the photosynthetic rate by almost 40% in the day and by 75% in the night. And this implies that during the night, photosynthesis was more dominating by the water deficit. Even after irrigation in the daytime, the photosynthesis rate was still lower from 8 a.m. to 4 p.m. On the one hand, supplemental lighting can increase the photosynthesis and make the plants grow faster. But on the other hand, it causes more transpiration during the nighttime, which gives rise to more water loss. And from the figure 2, we can see that the stomata of the non-irrigation group was thinner and shorter than the average among the irrigation region. A similar pattern was observed for pearl-wise and pearl-length. The SAM also showed that there was no significant difference in leaves grown under the control and the irrigation region, but that the stomata was damaged and thinner in the plants grown with non-irrigation region. So in this study, smaller stomata and prawns and reduced stomata density under the non-irrigation region are in line with the reduced stomatal conductance. The decrease in intercircular carbohydrate concentration during the daytime could indicate that stomatal conductance was the main factor limiting assimilation during the drought stress. The stomatal limitation increased during the early day and then decreased after 1 o'clock, suggesting that stomatal limitation to photosynthesis was dominating in the morning, but non-stomatal limitation was dominating in the afternoon. And from the table 3, we can see that there were no significant differences observed on chlorophyll content and carotenoid content between control and the irrigation region. However, plants grown with lighting but without irrigation during the night showed a significant reduction in leaf chlorophyll content. From the chlorophyll fluorescence parameter in the plants grown under the 3 regions were determined in the figure 4. The plant leaves with extended day lengths but no irrigation had higher FO and FM values, and the NPQ, a stress index, increased from 0.52 under the irrigation region to 0.74 under the non-irrigation region. Almost 30% increase occurred due to the drought stress. And the lowest robuscle activities were under the non-irrigation region were observed for initial, total, and maximal activities. So we can see that the chlorophyll fluorescence can provide a better understanding of the photosynthetic processes during drought stress in rose plants. If the excess energy under drought condition is not dissipated safely, overexcitation of the reaction centers of photosystem 2 can occur, which may limit photosynthesis. And in the present study, reduction in leaf chlorophyll content of the stressed rose plants may have accounted for some of the lower photosynthetic performance, and suggested that the reduction in the photosynthesis may be attributed to the damage inflicted on the photosynthetic apparatus of the photosystem 2. And the robuscle activity, the rate of the robuscle regeneration, and the rate of the process in the carbon circle were also limiting factors for the photosynthesis. The last part, conclusion. So from the study, we see that there were immediate depression of the photosynthesis under the non-irrigation region, but not under the irrigation region. There were both stomatal and non-stomatal limitations to the photosynthesis. Stomatal limitation was dominant in the morning, but non-stomatal limitation had further negative effects on the productivity by exacerbating the pigment content and biochemical limitations to the photosynthesis in the afternoon. And as a result, there was a decrease in plant quality and the cart rows during drought stress. Stomatal and non-stomatal limitations occurred coincidentally and were both important components for the photosynthetic drought stress. That's my presentation. Thanks for your attention.
Thank you very much, Dr. Liun Si. Now it is time for question. Okay, Professor Ki-Sung Kim.
Thank you. Nice to see you again, Dr. Shi.
Nice to
My
meet you
question is
again.
carbon dioxide. Do you supply the CO2 at night or daytime? Did you
Sorry.
supply the CO2
Pardon, please.
at night or daytime? Did you supply the carbon dioxide at night? The carbon dioxide, CO2.
Yeah.
What ppm?
In this experiment, the carbon dioxide was not applied in the day and the nighttime. It only investigated the supplemental lighting and the irrigation regime on the cartridges, not take the carbon dioxide into consideration.
I think if you had supplied more CO2, your results could have been much better.
Yeah, I will take
Thank
it
you.
into consideration. Thanks for your suggestion.
Okay, Professor Leon, good to see you
Yeah.
again, and thank you very much for your presentation. Okay, time is all, so thank you very much, Leon C.
Thank you, and so nice to see you again.
Okay, lastly, I'm so honored to be here as one of the steering members and serving a chairperson. Personally, I'm pleased to see my student. It is a wonderful time to me. Thank you very much for all. And now close session 2. I'd like to thanks all for your kind and for your some attention. Thank you very much. Thank you.
Thank you very much for Professor Wan-Sung Kim and all presenters today. Now this is the time for the poster presentation. And as you already know, we have opened our e-poster session in advance of this symposium. So for our online participants, we are now closing for all today's program, and please feel free to join the e-poster session through the website. And for tomorrow, business meeting will be started at 8 Korean time. And business meeting will be managed by Professor George Kanhoto, Chair of ISHS Division Plant Generating Resources and Biotechnology. Some important issues about the next venue and next convener will be discussed during the business meeting time. So please connect to the Zoom at 8 with the earphone or the headset, and tomorrow morning. So for the participants here, I'm going to have a further announcement soon. So thank you very much for joining us today, and please enjoy our poster session, and I'm looking forward to see you, all of you, tomorrow morning. Thank you.
you.